We're so excited at UCLA to host the inaugural Los Angeles Postdoctoral Research Symposium. And again, as I mentioned, this is completely organized by the 3 postdoc associations and many volunteers. So it's really impressive. It's also really impressive that you're here on a Thursday where there's really bad traffic, and it's like June gloom gray, and here you all are taking the day to be here. So kudos to you as well for making it here. As Johnny mentioned, this is a really unique opportunity for a number of reasons. And part of it is that you're all LA postdocs coming from different areas. And I'm supposed to do this, and I'm going to plug this several times. If you could today on social media, use the hashtag LA postdocs for those of you who are on social media to get the word out about this. But so how many people here are from Caltech? All right. USC? Nice. UCLA? Okay, you better be in the house. City of Hope? What? Okay, I'm sure they'll come. Cedar Sinai? Wow, okay. Children's Hospital? Yay! And then JPL NASA? Awesome, this is amazing. So we have 1, 2, 3, 4, 5, 6, 7. Well, we're going to put City of Hope in there because they're going to show up. So, I mean, there's 7 institutions. So, I mean, I've been working at UCLA for almost 13 years, and this is, not that that's forever, but in recent memory, this has never happened. So this is awesome. And again, feel free to use the hashtag and get the word out and be like, hey, lab mate who didn't come after lunch, or like come for the mixer in the evening or something. So there's a couple things that are going to be happening today. As Johnny mentioned, you know, you're here to sort of hear about cutting edge research. You're either presenting on it or you're learning about other people's work. So that in itself would seem to be enough. But the intent is more than that. So part of it is also to sort of make these connections and to foster a possibility of doing interdisciplinary research together or meeting each other. Or just, I mean, so many postdocs come from, or actually most of you do, come from somewhere else. There's very few LA natives that are here. So you're all not only coming here as cutting edge researchers, but also as sort of people who are probably pretty new to Los Angeles. And so it's also an opportunity to get to know each other. You already have some stuff in common, right? You're all postdocs, you're here, you're really smart, you have PhDs or MDs, all that good stuff. Okay, so again, I'm going to just plug the hashtag LA postdocs. And then I want to thank a bunch of people. So again, this couldn't have happened without the 3 postdoc organizations. The sponsors, CNSI, CTSI, were very generous in giving space and money. The panelists, the exhibitors, the judges, the planning committee, the different institutions, and leadership. And they're all, they're all in the booklet, but read through them and give a moment of gratitude to all of them for making this happen today. So now I want to get to the main event, which is the keynote. And we are absolutely delighted to have Dr. Paul Weiss here. And he is a graduate of, you know what, I'm at that age, this is terrible, where I need to get new glasses, so I'm like, I have to take it off to read. Anyway, okay, so graduate from MIT with all his degrees in chemistry, MIT with his undergrad and master's degrees and PhD in chemistry from University of Berkeley. He's a nanoscientist who holds the UC Presidential Chair and is a distinguished professor of chemistry and biochemistry, bioengineering, material science and engineering at UCLA, where he was previously director of this beautiful institute, the California Nanosystems Institute. He also currently holds visiting appointments at Harvard's Weiss Institute and several universities in Australia, China, and South Korea. He studies the ultimate limits of miniaturization, developing and applying new tools and methods for atomic resolution and spectroscopic imaging and patterning of chemical functionality. He led, co-authored, and published the technology roadmaps for the BRAIN Initiative and the U.S. Microbiome Initiative. He has won a number of awards in science, engineering, teaching, publishing, and communications, and just spend the whole day reading about that. But instead, I will also just mention he was a fellow of the American Academy of Arts and Sciences, American Association for the Advancement of Science, the American Chemical Society, and many other societies and institutions and institutes. And he's also the founding and current editor-in-chief of ACS Nano. So one more robust applause for this morning. Please join me in welcoming Paul.
Thanks so much. Okay. So thank you all for coming out so early. This is really a fantastic session that's been put together, and I think you'll find it very useful, not just today, but for the rest of your careers beyond postdocs as well. And we'll talk a little bit about that as we go. But I thought I'd start off here with a story that I tell my incoming freshmen and my incoming graduate students. And since you're mostly new postdocs to me, we can do this as well. So 2 people are in a train car going through the Alps together, and there's an artist and a scientist, and the artist is admiring the scenery, and the scientist starts to get a little bored. And then after about an hour of this, the scientist looks over at the artist and says, "Let's play a game. I'll ask you a question, and if you don't know the answer, you have to give me €1. And then you'll ask me, and if I don't know the answer, I have to give you €1. And the artist just ignores her, keeps looking out the window, and enjoying the scientist won't give up. She goes, "Okay, €10. I'll give you €10, but you only have to give me €1. Artist keeps looking out the window, so the scientist getting frustrated says, "Okay, €1,000. I'll give you €1,000. You only have to give me 1. So the artist looks over, nods her head. So the scientist goes, me first, me first. How far apart are the Earth and the Moon?" And the artist shrugs her shoulders and pulls out a euro coin and hands it over. And the scientist goes, "Okay, now your turn. You ask me something." So the artist thinks for a second and says, "Well, what goes up a mountain on 7 legs and comes down on 3? Scratches her head, and she pulls out a pad and starts looking through notes, and then she pulls out her iPad. They have Wi-Fi on the train, and she's looking through Wikipedia and the kingdoms and so forth, and can't figure it out. And after about an hour and a half, she pulls out 10 100-euro bills and hands them over and goes, "I give up." "What?" And the artist shrugs her shoulders and hands a euro coin back. Right? And so there are a couple of morals to this story. One is ask the high-value questions, and the other one is there's not always an answer. Okay, so keep that in mind as you're doing your postdocs and starting your careers, but we'll talk about these things as we go. Now, you're all in Los Angeles, so maybe you already understand what's so special about it, right? And one of the things that attracts me is there's stimulation from all sides at all times. And I thrive on chaos, and I thrive on stimulation, and so it's great for me. And I tell people, if that's what you like, come here. And you've all come here to see postdocs from across all different fields, so you're kind of a self-selected group. But if you really want to be left alone and work in your own lab, you probably didn't come today. You'd probably also be wasting your time coming to Los Angeles. Okay, but it's not only, right, movies and arts and so forth. It's really a center of creativity much more broadly. I'm sorry, I would have included Children's Hospital and City of Hope and JPL and Cedars. We work with all those institutions in various ways, and I've published with, I think, everybody but children so far. If you know LA Story, which is actually a documentary, not a comedy, if you haven't seen it, we would keep it for visitors in our lab to understand what LA is like. One of the things that we feel very strongly is that we're pushed to be creative, and if we don't do something crazy in our lab, we're not doing our jobs. And the advantage of that is that if it succeeds, people go, "Wow, that is amazing. How'd you ever think of that?" And if it doesn't work, nobody ever hears about it, right? And that's an important lesson for an academic career as well, is that the world is a peak meter. They're only going to see the things that work and the things that poke out above the clouds. And so while you will spend sometimes decades on a project, and we've done that, we have one going on 30 years now that hasn't worked yet, but we're pretty close, we think. We're on the, I think, 5th generation of graduate students on it, but we've done some things along the way. When that works, we think it'll be important. And there are other parts of our lab that have worked within a week, and others that we spent 20 years, and again, multiple generations to get going. But we've added new tools and capabilities through those efforts, and along the way, people have learned a lot, I would say. Okay, so you heard I come from Nanoscience. That actually didn't happen until my 2nd postdoc. It should also tell you something. I was trained in gas phase chemistry, stuff I would never even read anymore. My advisor got Nobel Prize the year I got my PhD, and I never did anything like that ever again. I've helped lead really the development of the field and also been an evangelist, as you'll see, and you're going to get a little bit of that right now about what some of the advantages of nanoscience are, but that is freely available to everybody. It's just our field developed in such a way that there's a requirement, essentially, that since we came from chemistry, physics, biology, medicine, engineering, toxicology, environmental health, and safety, that we learned each other's problems and approaches and applied those together to do new things and look for opportunities. And you'll see that, and we'll talk a little bit about the Brain Initiative and the Microbiome Initiative and why those came from nanoscience and not, for instance, from neuroscience in the case of the Brain Initiative. And maybe you'll just dial back social media a little bit when I say something unkind about that field, which I'm a neuroscientist in law, so I'm kind of allowed. Okay. So you might know that the National Nanotechnology Initiative began and funded our field to the tune of a bit north of $1 billion a year. Some of that was collected, money that was already being given out, but some of it was new sources. And at the 10th year anniversary of that initiative, we were asked as a journal, where we try and represent the field internationally, to say what we learned in the first decade, and if our funding were to continue, what we might do in the second decade. And that seemed like an important pair of questions to answer when asked by the people who were supporting us. Okay? And so we went back and reviewed the field, and you heard a little bit about what I do. What our lab is best known for is looking at the smallest switches and motors in the world. And so we look at molecules and assemblies inspired by biology, but those are too flexible for us because we want to go from quantum mechanics to mechanical engineering and experiment theory and simulation. So we use rigid synthetic molecules that we design to have functionality present or not present based on how we think these things might operate. And so in order to do that, we had to develop two capabilities in our laboratory. One was these microscopes that measure structure, function, and spectra, tens and hundreds of thousands of times of the same molecular assembly. And the other thing we needed to do was develop a chemical patterning capability where we could put molecules or even parts of molecules with precision down to 1/10 of an angstrom and simultaneously control chemical functionality out to centimeters. So really adding the chemical dimension to nanolithography. Okay, so that's where I kind of came from. Then I started that journal, and then I got, manuscripts from everything you can possibly imagine. And right now we get a little over 10,000 manuscripts a year. I read and rate every one of them. I'm behind by 2. By the time I came over here, I saw them come in just as I was closing up my computer. Well, something up, but not assigning them. And so we have 30 editors around the world, but I do get to see what's going on in the field, what people are trying to do. And then from 2007, when we founded the journal, till now, we're ranked, I think, 15 of all journals in all fields, and #1 in nano, #2 in engineering by Google Scholar, which is way better than Impact Factor, by the way. Look it up, you'll see Google Scholar metrics. Anyway, so what we realized was there are cases where we can make atomically precise structures. And there are times we should do that. For instance, to make contacts in electronics that have minimized resistance so your laptop doesn't burn you, so that a battery lasts longer, so you can do things faster, and so forth. But also, I would say the main thing we learned from this measurement of the switches and motors was that if you measure the same one over and over, you get a different answer. And when I say the same one, I don't mean the chemically the same one, I mean the very same one. And so we know the world, even at that precise scale, is heterogeneous. Many of you are in life sciences. How many? Of some sort, right. So we have a really big problem in life sciences, and that is our structural tools are all averaging, right? Crystallography, you have to make a crystal. I have a brother who's a crystallographer. He got his PhD across the way here a long time ago. Maybe, well, a couple of professors were born then, but most of the rest of you not. And #1, I don't like my crystals. start with that. But you only get the average in that you're looking at the periodic part of that. In NMR, right, you need a lot of molecules, and so you have an average. Even in cryo-TEM, which in the basement, we've gotten atomically resolved structures of viruses and so forth. There you're using 5,000 to 10,000 molecules or assemblies to get a structure and guessing the orientations, and you're getting an average that way. And so we really need tools that break that up. And in fact, the person who reinvigorated the Postdoctoral Society at UCLA was my first postdoc when I moved here in 2009, and has just got tenure in chemistry and biomedical engineering at Purdue, but Shelley Claridge. And she did something that I hope you will do, which is go around and learn what everybody does. And she connected with David Eisenberg, sort of king of amyloids over here. She worked in our group in scanning probes, went to the neuroscience community. We have several hundred neuroscience faculty and buildings and departments spread across different schools and so forth, and took on that problem of how do we measure biomolecular structures without averaging. And she was able to measure one in 2 years. That turns out not to be something useful where you can then measure 1,000 of them and relate that to function. But then by collaborating with the mathematicians here, we're able to accelerate that down to 10 minutes. So it's almost the entire acceleration of the genome project, not just in our lab, but at least with us as a hub, grabbing different collaborators around a campus and around the world. Okay, so heterogeneity and perfection, that's one thing, but I would say one of the things that in our field we try to do, and I'll be more specific about this, is in the whole biotechnology revolution was enabled by new tools, and particularly automated DNA sequencing, DNA synthesis, protein sequencing, protein synthesis. And those, again, were related all to one person who realized those were needed, and he established collaborations, and that was Lee Hood, who I spent a year with about 10 years before I came here, when he was still at University of Washington. And we'll talk a little bit about how he thinks and why I took 6 months to figure it out, what he was even talking about, and then why that kind of thinking is useful, especially at this point in your career. Okay. And so because the way our field has developed, we kind of naturally have that communication, but that is open to anybody, and you're going to learn that here and with each other. And I particularly want to encourage people in life sciences and not in life sciences to cross-pollinate. When Shelley pushed the Postdoctoral Society here, I thought the most interesting part of it was the life science postdoc saying, well, I'm in my 6th year, right? And the physicist saying, 6th year, right? You'd be halfway to tenure. And there's a very different lay of the land in terms of publications, career trajectory, what you pick up, how in some areas you dissociate from the previous groups, and others, you work with your mentors until well after they pass away. In the case of my wife's, career trajectory, there are experiments that our son fueled with martinis at SFN when he was 11 years old, and her advisor passed away a couple of years ago, and they're still named, those experiments still named after him, and in collaboration with. Okay. All right. If you're interested in those views in nanoscience, we published those here. I'll point out a couple of other things special about LA and that and so forth. So when we took on a problem in chronic pain, one of our key collaborators was a guy who didn't graduate from high school, John Stevenson. He wrote and directed Kung Fu Panda. He used to fly back from London to work with us every couple of weeks because he was interested in the topic, and because we learned that chronic pain is more about learning and memory, and in the case of trying to treat it, we thought distraction, than it is about injury. And he brought to us source codes of games and so forth that we could modify, and he taught us about perception and about how when you make a movie or tell a story or put together a game, you purposefully put effort and intensity into different dimensions that we don't even think about when we give talks or write papers or write a proposal, but would do us a lot of good to know. And so he was phenomenal at helping us design experiments and bringing in other people. You also get the opportunity to play sometimes, so I helped with about 10 or 12 projects. The best one known as the Terminator Genesis, which I don't think anybody saw. But In the building here, we have Science Entertainment Exchange, and if you have a chance to engage with them, it's a collaboration with the National Academy of Sciences and the Directors Guild, which is just down the street. And so people from the entertainment industry who want to promote science, they don't want policemen for science, but they want ideas and creativity and, what have you got that we didn't have in a previous movie in the series, or, a game people are trying to develop, or how could you put something in a game that might be useful, equivalent to protein folding. All those things are opportunities that can arise if you keep your eyes open for them, and I've had several postdocs in the lab also help. Okay, so let's talk about 3 people. We lost Millie Dresselhaus. She was called the Queen of Carbon, and she was a nanoscientist before the field was there by decades. And she had some serious difficulties during her graduate and postdoctoral and academic career as follows. I did an interview with her that you can read here. When she was a graduate student, she was in physics at Chicago. Anyone come from University of Chicago? Not so many people make that move. The one physics thesis advisor there didn't think women should get PhDs, and so he wouldn't help her. And so Enrico Fermi adopted her and brought her over for dinner once a week. She was the same age as his daughter, and promoted her career. When she went to do a postdoc at Cornell, her husband was also a physicist. Cornell and many places had nepotism rules at that time that if one part of a couple was hired, the other one could not be hired by the university. So while she had an NSF postdoctoral fellowship and her own funding, she wasn't allowed to have a mentor at Cornell and had to work on her own. When she went to MIT and worked at Lincoln Labs, further nepotism rules came in, and she was told she had to be in by a certain time of day. She had 4 kids, and then the Rockefellers came in and established a professorship there that let her move over to the academic side, and her career took off from there. But she has a lot of interesting things to say about, she worked in carbon nanomaterials before they existed and looked at what their properties would be. And when that field came into being, even before that field came into being, she was known as the person who knew the most about those materials. And then many of her predictions, and then all the people that she inspired and mentored came into prominence in that area. And, a lot of that is decades of saying, I think this is interesting and important, even if the materials don't exist. Okay. Lee Hood I mentioned. So he had the one thing I want to say about him, he had this weird way of talking where he didn't distinguish between the past and the future that he knew was going to happen. And so he would talk about some result or experiment, and I'd say, I missed that one. He said, well, it hasn't been done yet. Yeah, I'd say, where's the paper? He goes, hasn't been done yet. I'm like, and eventually I realized what he was doing is he was just assuming everything that could be done was already done, and then saying, what should I do next? And it's like a chess game in that way, where you think of all the moves that have to happen, and then you go forward from there. And when things were too slow in terms of technology development, he would just take it on himself, and that ended up being very significant. So all those, all the underlying tools of biotechnology revolution really came out of his impatience to a large extent, and he would never talk about instruments. He came to my previous institution, gave 5 talks, showed 1 instrument slide, and then I was the only one who brought him to my lab. Otherwise, they had him sitting in some office where people would come in, have these kind of sterile conversations. And when you do that, you also have a lot of impact on the world, and that is tremendously possible to do in all the places you are. There's a really vibrant entrepreneurship community that's growing very rapidly here in Los Angeles. And if you are interested in that, I'm going to be around during the breaks and so forth, but this is one place you can come, Caltech and all the hospitals and other universities have programs that are growing very quickly. The third one is a guy who comes from my field, George Whitesides. I actually had him for freshman chemistry, organic, actually. I never took freshman chemistry. Took it pass, fail, passed. And he's mentored just a very large number of successful academics and others. And he has this matrix of saying, well, look, when you start your academic career, or I would say, grad student, postdoc, even undergraduate researcher, choose something you think is important. His view of it was, well, if it works, of course, you're golden. If you chose something unimportant that works, who cares? If you chose something unimportant and it doesn't work, then you're dead. But that off-diagonal term of you chose something important and it didn't work, you get credit for identifying the problem and establishing some paths, or maybe paths to avoid, towards that problem, but you also become the expert in that area, and that's an opportunity for you. And so when you go on, both in your postdoc and when you go on to what's next, by all means, choose something you care about and you can't wait to get up out of bed and do in the morning. And there's going to be a divergence in this community where some people will, continue projects that they worked on before, and others are essentially by field forbidden from doing that. And so talk to each other and see which part of that community your field is. Okay, so a little more background. Once upon a time, I was the first one to move atoms around on a surface, showing we could look at we could measure not only structures with the microscopes we built, but we could assemble these perfect structures. This was the microscope where, after I left, the guy I worked with, Don Eigler, learned to spell and wrote at IBM. But I just wanted to find out what was underneath the atoms, and that was I was sliding around. And so that added a whole set of capabilities to what we could do. And to me, the most important image that was ever recorded was actually a pair of them, where on this one semiconductor surface that has two different elements, one, if you remember back to high school and freshman chemistry, it's more electronegative than the other. Sorry, I teach freshmen, I just can't get past this. One's more electronegative than the other, so there's charge transfer. So when we look at the electron orbitals that are filled, we see the more electronegative one. When we look at the orbitals that are empty, by having electrons go the other way, we find the ones that are less electronegative. And then in that way, for me, what I saw is here was a way to look at a surface the way a molecule or part of a molecule or an atom would. And if you remember, electrophilic and nucleophilic, those functional groups or atoms would go to different parts of the surface, and we could essentially put on goggles and look at the surface the way a molecule would. And that's what got me excited about this technique that was only recently developed at the time that I started working on it. I mentioned perfection, and it is possible to make precise structures. These are all carbon graphene nanoribbons that are sort of devices in line, and we do some of that, and there's a Berkeley group that does some of that. But the more interesting things turn out to be the heterogeneity. And so early on in this field, I'm just going to give you about 2 more slides of data, we had developed a new microscope. And that microscope could look at insulating surfaces. And the reason we did that was IBM and Bell Labs at that time dominated semiconductor surfaces and worlds. And they even had to negotiate between adjacent labs. You look at this material and I'll look at that material. Or even the same material but different surfaces. And then some academic labs looked at metals, mostly catalysis and corrosion. I wasn't very interested in that. But there are a tremendous number of interesting insulators in the world, including most of biology. And so we developed a microscope that could image with atomic resolution and record spectroscopic, for me, that meant chemical information. We're looking for as big, as wide a set of subjects as we could find. And this guy, Dave Alara, back when he was at Bell Labs, this is him looking very unhappy at his 80th birthday, with Ralph Nuzzo, had developed these monolayers of molecules that would form chemical bonds to substrates, ordered layers, and they were very popular in what was thought to be the gateway to chemical patterning at the time. Whitesides had been Ralph's mentor, and he picked that up from Ralph. David moved to Penn State, where I started my career and was developing this microscope. And at the time, it was thought these molecules went down randomly and irreversibly and were immobile on the surface. And so Dave suggested we put down 2 different molecules and use our spectroscopic capability to tell them apart. That sounded like a good way to test out the microscope. You could use all kinds of different molecules for this. And at that time, George had published a paper with a guy named John Deutsch. You never hear him? He used to be called the disgraced former head of the CIA, but it's no longer a unique identifier. So his last scientific paper said you could never get phase separation in these systems. And our very first images showed you did. And the false assumption that George and John had made was that these films were always at equilibrium, and they never were. And so all that led to our ability to pattern molecules on surfaces, to control defects, even to place individual functional molecules, like it's already June, so they're starting to play Love Actually for Christmas. Right? The Bill Nighy character, right, surrounds himself by this matrix, like we would put a functional molecule into a domain boundary and with our microscopes measure its function, measure its spectra, measure its structure, and so forth. Okay, well, and we did that, and I'm going to skip all the rest of it. But this came to the attention of a neuroscientist who was trying to develop sensors to put in the brain to listen in on chemical communication. And she was my colleague at Penn State at the time. We'd stolen her away from NIMH, where she'd been a student, postdoc, staff fellow, is that what they're called, and then senior fellow, and so forth. And she said, why don't you give me my favorite neurotransmitter, serotonin, on surface, where I can capture molecules involved in neurotransmission in the brain, receptors and transporters, and develop artificial receptors that we can use to listen in and understand what a thought is, what a memory is, the difference between function and malfunction. We started working together 17 years ago, and tomorrow will be our 14th anniversary. She's coming back from NIH, where she's at the director's meeting. She won this Caro won Director's Award last year for this project. This paper where we finally made that work 20 years into her efforts, 16 into our collaboration. We probably put $3 million into that paper at a minimum in all that time. And we did not have federal funding until basically we'd made it work already. We stole every bit of our cheer funds, our setup funds, anything we could do in chemical patterning to make that happen. And that ended up being the basis of the chemical side of the Brain Initiative. So I was asked to put together a committee on nanoscience for the White House, saying, come up with a big challenge. And the computer scientists had done this very successfully, and they didn't get a lot of attention, but for nanoscience, they wanted something that would get attention. And we agreed that since, coincidentally, the scale of the semiconductor technology that's in use in your laptops and phones is the same as the synapse scale, let's see if we can somehow merge those efforts and figure out how neural circuits work in the brain. And so the problem that we had was most of the committee was physicists and engineers who said the brain's a computer, measure the voltages and you're done. It's not, by the way. It's the neuroscientists here. And so we fought, saying if you don't measure the chemistry, it's not worthwhile. And we fought that out in literature over a couple of years. And then, as you heard, we published the technology roadmap a couple of weeks before the president's announcement. He changed the name the night before, so the names don't match exactly, but that's okay. Now, right after that, I'll finish up with this. The White House had another initiative that came from another group called Precision Medicine Initiative. Oh, and I'll point out the neuroscientists did not support that effort. We had to go sit on people to get them to shut up, right, to let that initiative go forward. For instance, the head of SFN. And even Obama played a role in pulling in people who we thought might be antagonistic and made them head of a study about what we would do. He was sort of in on what we needed to do. So Precision Medicine Initiative, many of you probably haven't heard of, and the reason the White House decided that it hadn't worked out as well as the Brain Initiative is that the Brain Initiative had been led by nanoscientists who brought in people from other fields, including people who'd led the Human Genome Project, including neuroscientists, including engineers who could come up with ways to get the information out. And so even though the microbiome is arguably less nano than the Brain Initiative, we were asked to lead that one as well with that same idea in mind. And that was the third and final big initiative that came out of OSDP before life changed. Okay. I'll point out around here, by grabbing dentists, right? Oral microbiome is much more accessible than gut microbiome as far as I can tell. And so that seemed like an easier place to score using some of the same technology we developed for the brain, but to listen in on important chemical signaling molecules. And that led us to other work that we can talk about that spinning out in the companies now here based on what we learned from those conversations with, when you're in the dentist, you don't talk very much, but with friends who we sit down for coffee with, and that's happening again later today, we learned what the problems were and came up with some simple solutions that turn out to be very useful for about half the population over 30 in the U.S. So pretty good opportunity there. Okay. And this stuff I'll just talk about later. We put together teams. Another person who saw chemical patterning as useful was a clinical fellow in my lab who recently joined the pediatrics faculty. And we opened up new opportunities in high-throughput gene editing and in 6 different strategies: no viruses, no electroporation, no harsh chemicals. All 6 worked within a week. And that's never happened in my lab. And that brought in a bunch of MDs and MD PhDs that has sort of taken off. So I think what I'll do is I'll just stop there, and I will be around for, oh wait, no, I have one other thing. Yeah, so I will be around, you can read. I'll be around for breaks and to talk, but I really encourage you to grab each other, and especially people who are in very different fields, and ask them what's expected of them. And you'll, especially some of the people in hospitals may not see the physical scientists and engineers as much. Caltech, less exposure to medicine maybe, unless you do the City program or something. But find each other and explore as you heard in the introduction. That's a real opportunity you have here. And keep touch and visit each other's labs and learn each other's problems, and you may find an opportunity to do something really interesting together. Thanks.
Good morning, everyone. My name is Nasim Ferdos, and I am a postdoc at USC Roybal Institute on Aging. And I'm a social scientist. I am a health economist and a health services researcher. And the research I'm presenting today is about the evolution of nursing home industry in states with different certificate of need policies. So it's simply, in this research, I'm simply showing that how one policy can influence a nursing home market, in a long run during like 24 years of study, and what happens in terms of the quality of care and supply of care based on one small policy. This policy is a certificate of need policy, which is a policy that requires states for expansion of hospitals, nursing homes, and home health agency. So they need state approval in order to expand or in order to enter the market. And it was one of the earliest efforts to curb the needless healthcare spending in the US, and it was established in the early 1970s. And it was initially designed to limit and manage the expansion of different healthcare services, healthcare institutions, to control healthcare costs. And it was the rationale of putting such a policy was that because policymakers believe that because there is not much competition in the market, so the prices are higher, and hospitals and nursing homes charge very high prices. So we put this policy to avoid duplicate costs and this kind of things. So, and at its peak, almost all states except Louisiana had some sort of con policies in effect. But in terms of the policy, economic modeling, it is seen as a supply-based policy that controls the expansion of services, and it can be seen as a barrier to entry for new nursing homes, for new hospitals. It's not easy for them to enter the market. So it's like a barrier to entry, and barrier to entry reduces the level of competition in the market. There is not enough competition, and that's why it may influence the quality of care in this setting. So we want to see what happens to the quality of care in the long run. So in the nursing home market, I'm specifically looking at the nursing home market and home health market. And in the nursing home market, Con programs are mainly targeting the long-term care because they're targeting nursing home and home health, which provide long-term care to the patients. And the long-run presence of con policies raises the important concern of a barrier to entry and lower competition in the market, as I just mentioned. So nursing homes in states with and without con may evolve in very different competitive markets. And because not all states have the same policies, so we can compare these different nursing homes in different markets or different states to see what happens to them in the long run. So we want to see what nursing homes do in different competitive atmospheres in the long run. So the objective is to examine the evolvement of nursing homes, and by the evolvement, I'm looking at the supply of services and quality of services, what happens to them during the study years. So I study nursing homes in all US states between 1992 to 2017, but I specifically look at the states which have not changed their con policies during the study years. So if they had a policy, so it hasn't changed during these years. And I use the OSCAR data, which is the most comprehensive data for long-term care services in the US. And the con regulation data was obtained from National Conference of State Legislation website. So among the 44 states in the US that have not changed their con policies during the study years, 19 states had only nursing home policies in effect during the study years. 15 states had both nursing home policies and home health policies, and 10 states didn't have any con policies in effect. So it's a good, we can have a good comparison because we have enough states in each of the groups. And the variables that I'm looking at are categorized in three groups: the nursing home structural characteristics, which I am looking at the occupancy rate of nursing homes, the percent of Medicare paid patients, and the number of nursing home beds. And it's important to look at the Medicare paid patients because in nursing homes, most of the funding comes from state funding for Medicaid and Medicare patients. So it's important to look at the percentage of these patients. And I also look at the nursing home staffing mix, which is kind of like a quality measure. I look at the direct care hour per patient per day and also the ratio of registered nurses to nurses because they both show a quality of care in each settings. And I also consider some MDS-based nursing home quality measures, which are the share of patients with bed sores, share of patients receiving antipsychotics, and the share of patients who were physically restrained during the study years. And for the analysis, I do the analysis based on the nursing home fixed effects. So we consider the quality or the differences between in nursing homes. So we consider those effects. And we estimate them separately for each type of state. We estimate the regression separately. And today I just showed the plots because it's easier to see how they are affecting the market in the long run. And for the plots, I show the growth plots. So these are plotted based on the year 1992. So everything you see is that like how each of these variables have changed or have grown during the study years. And the first, these three graphs show the structural characteristics of nursing homes over time. And the red ones show the results for states that have not, that didn't have any con policies in effect. The blue line are the states with only nursing home con policies, and the green ones are the ones who have both nursing home and home health con policies in effect. And the graph, panel A on the left shows the occupancy rate. And as you can see, occupancy rate have declined in all states, in all the three types of states, but it has declined more rapidly in states with with nursing home policies. And the one in the middle shows the share of Medicare paid patients. It has increased in all three types of states, in nursing home in all three types of states, but it has increased more rapidly in states which have some sort of con policies. So it means that they have more Medicare patients, which means that they need more care, they need more registered nurses, they need more amount of or time spent for each patient. So we'll see if they have had that. That's a measure of quality. And the panel C on the right shows the supply of beds that we can see. It has declined in states with no con policy and declined in states with nursing home con policies, but it has increased very rapidly in states with home health con policies. And this shows the nursing home staffing mix over time. And as we can see, the left one, the direct care per hour per patient is not much different among the states, a little bit higher in states with without con policies. And the right one, panel B, shows the ratio of registered nurses to nurses. And as you can see, it's the lowest in states with both nursing home and home health con. And as you saw in the previous graph, they have increased the supply of beds, and the share of patients with share of Medicare patients is higher in these states, but they have lower registered nurses. So which means that probably the quality of care is lower in these settings, in these nursing homes. And this last one shows the growth in nursing home quality measures, which is the left one shows bed sores, not much differences, not a significant difference, but a little bit higher in states with con policies, which we could expect because they have less registered nurses and the time spent for each patient is lower. The use of antipsychotics, the ratio of patients receiving antipsychotics have increased in all types of states, but it has increased more rapidly in states with some sort of con policies. And the ratio of restraints have declined in all states, but it is a little bit higher in states with con policies. So to wrap up, the number of nursing home beds constantly grow in states with home health con, while it declined in no con states. And home health con law, which were mainly set to influence home health industry, have affected nursing home industry as well. And the constant growth of nursing home and nursing home bed supply alongside a decline in number of nursing homes shows that there is a barrier to entry and there is market power. And this market power, as we expected, has negatively influenced the market. So states with the con states have increased their share of Medicare paid patients, but they lag behind increasing registered nurses, and there's no difference in terms of the amount of care the patient receives. So this means that they have to increase this. So this means that the quality is lower. And to conclude, this study provided evidence on how market power can negatively influence the nursing home market, and we found that nursing home quality measures improved more rapidly in states without con, and the market power negatively affects the quality of care in the long run. And thank you.
Thank you very much.
Hi, everyone. I would like to talk to you about life, its origins, its definition. So to start with, I'd just like everyone to take a quick moment to think about what your own definition of life is. What separates life from non-life? Is it the molecules that it's made of? Is it what it does? Is it its presence in an ecosystem? There's no correct answer, but I think we can all gain something from thinking about this every now and again. So my job is to try and understand where life came from and where it might originate elsewhere in the universe. And I like to approach this problem from a theoretical perspective. And today I'm going to present my own definition of life, which is based upon information processing, computation, and complexity. So when I say information processing and computation, I basically mean processes where you have information being put into a system, I mean, in the most obvious case, we put information into our computers in terms of an input of some form, and then the computational processes manipulates that information and changes its form, normally into a form which is somehow useful, has some kind of function, and it's then output somehow. And I'm going to argue that life is performing computation, it's performing information processing, and in fact, this is not the only condition, but the most important condition which separates it from non-life. So often when we think of computation, we think of things like neural networks. Some of you may be neuroscientists. But in fact, in the last, especially since the systems and synthetic biology revolution, we're now discovering that life performs computation on all kinds of different levels. So this is a diagram of a bacteriophage. It's been shown that actually bacteriophages communicate and can make decisions based on what environment they find themselves in, whereas you would expect that, I mean, most biologists consider that viruses aren't even alive, let alone able to make decisions. This is a picture of a bristlecone pine. These are some of the oldest organisms in the world. You can find them not too far from here, a little bit north in California. They actually, their seeds are preferentially released when there's a forest fire. When there's a forest fire, of course, most organisms are damaged or killed. But these guys have been around so long that they've learned through an evolutionary process that in the conditions of a forest fire, your competition is probably going to be reduced, and so now might be a good time to proliferate if you can. So I'm going to just take you through a few different levels at which biological systems compute. So most of us are familiar with computation being performed by computers or our own minds, our brains, where you have, in the case of brains, you have neurons which are actually performing the computation, perhaps doing some kind of nonlinear some kind of summation over inputs and then generating an output. And the information is communicated via the synapses and the microtubules. Again, with computers, you have electron flows communicating the information and the chips performing the computation. You have logic gates. But we can look one level deeper. So this is a single-celled organism called Lacromaria. And if you watch this YouTube video, What Bodies Think About Bioelectric Computation, you will see that this single-celled organism in real time will swim around, seek out targets. It's actually a single-celled predator. So it's taking measurements of its environment, making decisions based on that information. So even this single-celled organism does quite sophisticated computation. And in fact, a lot of the communication of that information is through charged particle flows. So this idea of computation through the manipulation of electron flows or ion flows is actually very ancient in life's evolution. But we can go even further down. So this work, starting in the mid-90s, proposed that even protein molecules, like these two molecules here, can be thought of as computational devices. The one on the left here actually acts like an AND gate. So depending on which ligands it's binding with, it will change its conformation and produce different outputs as a function of its inputs. And likewise, there are various molecular switches that you can find in the biological repertoire. And I recommend having a look at this book where he surveys a whole range of ways that life performs computations. And in fact, with recent advances in the last 10 or 20 years in the area of thermodynamics, stochastic thermodynamics, and computational mechanics, we can actually make rigorous measurements of the degree to which a system is performing computation. And what we do is we basically take measurements of the system, turn those measurements into a symbolic series, like a binary series, for example, and then you can generate you can generate a minimal, optimal Markovian model that can reproduce that time series. And from that model, you can then calculate the information content that's contained in your signal and how much computation is happening within that signal. And this method was pioneered by Professor Crutchfield of UC Davis. There are a few examples here. So for very simple systems like the one-sided coin, you just have one state, one transition. For a fair coin, you have one state and two different transitions. And this, from these models, you can calculate the rate at which entropy is being generated, and you can calculate this metric called the statistical complexity, which actually measures how much information is in the state space of this model. For the slightly more complicated model on the far right, it has a statistical complexity of 1 bit because the state space of the model stores at least 1 bit of information. Now these of course are highly simple examples, but the method generalizes. And we're now applying that technique to the analysis of signals from planets. We're starting with planet Earth because we have the best data on planet Earth, and basically looking at variations in light levels. When we just look at the Earth and pretend that we're looking at it from a very great distance, and it's just a distant dot of basically 1 pixel, and we have time series of different wavelengths of light, we can analyze the information content of those time series, and you can see, for example, the Earth's rotation and seasonal variations. And so in the end, when you do this process of epsilon machine reconstruction, you can calculate these complexity metrics, and our theory is that planets which are most likely to host life will be the ones that have the highest values of these complexity metrics. And we also do work on chemical reaction networks and looking for computation in chemical reaction networks. And if you want to hear more about that and other projects, please come and see me afterwards. For the sake of timekeeping, I'll leave it there. Thank you.
All right. Well, hello, everyone. Just as a quick introduction, so you know my name already, but I'm here at UCLA, got my PhD at VCU, and I'm primarily interested in networks. And I started approaching them from more of a biology direction, studying protein-protein interactions, and those fit really nicely into networks because you can say, okay, this protein interacts with another. But lately I've been approaching them from more of the direction of data integration. And I'll get into that, what I really mean by that, shortly here. For example, I'm just one researcher out of the many out there, including everyone here in the audience. And this year I hope to publish a few papers. Maybe 3 would be nice. A few more would probably be better. And then, of course, everyone else in the field, or even in different fields, will hope to publish their own papers. And we know that, despite the fact that we have many of these papers out there, they're going to be similar in certain ways. They're going to have conceptual similarities. They're going to have conclusions that perhaps agree or maybe even disagree with each other. But those are still similarities that we want to be able to organize. We want to be able to be aware of them. And that's a really challenging problem, particularly in biomedicine. Because if you look at PubMed, kind of your primary source for biomedical literature out there, there's 29.7 million records in there. So each of those links to one publication generally. That's a lot to have to read. Since 2014, they add more than 1 million records every year. That number's only growing. And that even includes more than 2 million clinical case reports. So each one of those documents is description of at least one or more patients and the kind of disease presentation or the symptoms or the particular medical procedures that they went through. So we not only have experimental records in this set, but we have clinical observations as well. So different kinds of information, different kinds of documents, and a whole lot of them. But that really pales in comparison to the scale of electronic health records and medical records. Even in the US alone, if we look at all the medical visits and all the records that they generate in a particular year, we're talking about more than 1 billion records every year. You're not going to read all of them. And even worse, these records are often particular to one location and medical system. They might not be particularly interoperable. And if you try to do something that seems as simple as assembling one patient's entire medical history across their entire life, that is a very challenging problem, not just for interoperability, but due to the total number of documents and records that you may have to integrate. So we're dealing with a couple different kinds of problems, many of which come down to them being unstructured. These are documents that vary in content and format. More specifically, they don't neatly fit into databases. We're not talking about the kind of things like a gene sequence or a genome where we might be able to say, okay, this comes from this particular species. We know, the A's, C's, G's, and T's. We can fit them into our database and then align those with other things. We just can't do that with this text data. It is largely unstructured. And with biomedicine in particular, it contains a lot of technical language, domain vocabulary, acronyms, things that you need a little bit more of a domain expertise to even begin to interpret. So kind of adding to that problem of being unstructured. So why is this more of a problem in general? Why do we care? It's a lot of data. It contains valuable knowledge. And if we seek comprehensive experimental design and understanding, if we actually want to be able to design experiments where we know, or as close as we can get to of a fact, that what we're looking at is the whole picture, we need to know what's already been observed. We need to know what's already been done and perhaps even published. We want to be able to connect the clinical observations that we see in things like electronic health records with experimental findings. In some instances, that means as simple as realizing, okay, we have a particular protein, it's involved in a particular disease or maybe multiple diseases. It's also involved in particular biochemical processes. These are the kind of things that we know we need to do for each individual experiment, but we also want to look into the literature and say, okay, what's already been done in some of these instances? So what's stopping us? Many of these data sets, as I mentioned, are just too large. EHRs are fragmented and not interoperable. We need a lot of accuracy and precision to be able to make conclusions based on them. But the methods for dealing with a lot of this data integration and mining are far too technical and require adaptation for biomedical language. But the status quo is really strong in working with these things as well. The status quo in some cases meaning just a Google search. If my question is, what are the proteins involved in mitochondrial disease? So these are systemic diseases, often genetic. If I type in that, I get more than 47 million results, which I'm not going to read all of. I'm just going to skim off the top and assume that those are the best results. That's how Google's designed. But even if I look at something like PubMed, I punch in that same search, I'm going to get more than 6,000 results, of which I'm probably not going to read all of them either. I need to carefully design my search to realize, okay, I'm just going to zoom in on one very specific aspect of here and maybe get a manageable number of documents that I, as a human, can read. It may go as far as realizing, okay, I need to actually go into the lab and try to answer this question myself. But even then, I'm doomed to possibly repeat the past and eventually realize, oh, a lot of these observations have already been made. That might be okay, but in terms of what my career looks like, I might spend my entire duration of my career just trying to answer that problem. So I think this is a job for computers. Now, in biology in particular, we're actually not really not alien to the idea of relationships in between documents, and we can even assemble them into entire networks like that. But when we're looking at things like a patient's experience in a particular disease presentation, assembling them into networks is a really useful thing to be able to do. We can assemble it on the basis of the things they were diagnosed with, their diagnostic results, the drugs they were treated with. Then we can connect those drugs to particular things that we know about them, this domain knowledge from databases. We can do the same for their disease presentations. And then perhaps most importantly, we can connect that through to other disease presentations and realize similarities between them and the diagnostics and the treatments that those patients experienced. We can also approach it from a little bit more of a protein-focused direction and realize, okay, for a given disease here, mitochondrial complex 5 deficiency, so a mitochondrial disease, we can realize some of the proteins that it's involved in and then connect those through to the specific patient presentations that we saw of that disease as well and hope to find some similarities. This really becomes useful once we start expanding out to much larger networks, potentially thousands or tens of thousands of patients, or much larger depending upon the amount of data that we have available. So how do we actually get that structured data? Well, sometimes as simple as just doing some named entity recognition or relation extraction, simply just breaking down the text into its component parts, but we do need to bring in that domain knowledge as well. So that's exactly what I and our group has been doing. We have this set called metadata acquired from clinical case reports. And that's exactly what it sounds like. We started with a bunch of published clinical case reports, about more than 3,000 in total, and that's all published and freely available as a data set. But we're going into more the direction of building systems for automating this process and doing some biomedical information extraction, getting a handle on the availability of the data that we have out there. Because really we want to do some natural language understanding. We want this to be fully a job for computers to assist clinicians and researchers with bridging that gap between experimental results and clinical observations, so they don't have to read 6,000 results out of a single PubMed or even a Google search. So with that, I'll thank these folks and take any questions.
Good morning, everyone. I'll try to give you an overview about the work we are doing at JPL in terms of radioisotope thermoelectric generators. When we want to do science in the deep space, solar is not a really feasible technology that we can use to power, to power spacecraft, especially because getting away from the sun, the solar irradiance is going to get smaller. So to get the same amount of power, you're going to need way bigger solar panels. So this generates a big lean for the spacecraft in terms of weight. Batteries are characterized by relatively short lives, and when we want to go at the end, for example, of our solar system, we take years just to get there before being able to start the science we want to do. So in all these situations, thermoelectric generators are probably the key technology to be used for powering the spacecrafts. Since 1961, who's the year of the Transit 4 missions, NASA used 41 RTGs successfully on 23 spacecraft. So far, no mission has to end due to the failure of a radioisotope thermoelectric generator. They're extremely reliable devices. They have no moving parts, and they can provide energy for a lot of years. For example, just think about the Voyager missions 1 and 2 that have been launched in '77, and they still have RTGs operating and giving power to explore what is existing just outside our solar system, because if you heard, like in 2012 and then Voyager 2 last year, they entered the interstellar space. So they left the area of our solar system when you have gravitational forces coming from our sun. And so Voyager 1 became the furthest human-made object traveling in the universe. This is the architecture of EMM-RTG, so enhanced multi-mission thermoelectric generators. The core is present the heat source. It's plutonium oxide, isotope 238. The natural decay of this isotope generates heat, and by the time it travels to the hot side of the thermoelectric module, we have a temperature of 1373 K. The cold side of the thermoelectric module is kept at 473 K approximately. And here we can see where the thermoelectric module is located. In this specific configuration, we have 768 thermoelectric couples that converts the heat from the heat source directly into usable electricity. Here we can see a picture from Curiosity mounting the MMRTG, who's a previous version of the generator I just showed you. And yeah, this is a very nice selfie coming directly from our rover on Mars. Thermoelectric modules are formed by PN junctions that are thermally connected in parallel, electrically in series. And as I said, they transform heat into electricity or electricity into heat. Two main effects exist in thermoelectricity. On one side, we have the Seebeck effect used for the generations, what is used in the RTGs. So when we apply a temperature difference at our PN junction, this is going to induce a flux of charge carriers that are electrons in the N-type and holes in the P-type from the hot to the cold side, and the cold side generates potential. The potential in the Seebeck effect is proportional to the Seebeck coefficient, the difference in temperature at the junction. Thermoelectric coolers instead use the Peltier effect, where basically a current applied to the junction generates still this flux of charge carriers, and also a heat flux moving from the hot side to the cold side, so cooling down the top surface and releasing the heat at the bottom of the junction. The heat flux generated is proportional to the Seebeck temperature, so the Peltier coefficient, times the intensity of current applied to the junction. The efficiency of the generator is a function of mainly three parameters: the temperature of the hot side, the temperature of the cold side, and the ZT, what's called figure of merit, and is a dimensionless parameter used to compare the quality of thermoelectric materials. To have efficiencies that can go above the 20%, we would need an average ZT of about 2. But actually today, the state of the art of materials are about 1.1, with a peak value of ZT around 1.5 or 2. Figure of merit, as I said, is defined by the Seebeck coefficient, alpha, electrical conductivity, absolute temperature, and the thermal conductivity. So to have high ZT, we need high Seebeck, high electrical conductivity, and low thermal conductivity. But as you can see here from the plot, all the three parameters are very strictly interconnected. So the optimization of one of them generates worsening of at least one of the others. So the main challenge for thermoelectric material research like myself is to find a way to decouple the optimization of these parameters, and aiming to find materials that can transfer heat as a glass, so they're thermal insulator, but at the same time they can transfer electrons or charge as a crystal, so they're very conductive. The strategies we're using at JPL to improve the ZT starts with looking for a complex crystal structure that can have a lot of atoms inside, a lot of defects, so we can have low thermal conductivity. Once we start with this material, we can then manipulate the electronic properties, doing band engineering, so alloying and doping. We can tailor the band structure in order to improve electrical conductivity. With compositing, we observe that we can improve the mechanical stability of materials while increasing conductivity, at the same time reducing the thermal conductivity. But everything is really non-trivial to figure out which kind of materials we want to use. We try to couple simulation and experimental. So with simulation, we try to guide the experimental, then we do like a semi-empirical modeling to optimize the material system. At the system level, we use segmentation to improve the average ZT. So each lag is not composed of just one material, but of several materials, let's say, for example, 2, in order to use each material just in the temperature window when they show their best efficiencies, and overall, the total efficiency of the module is going to be improved. Larger improvement has been done in the last 15 years. If we consider like GP-HS RTG that was used in the 80s, had efficiency of 5%, and then we move up to today, and we have a next generation RTG that has an efficiency of about 15%. The materials are currently used is on the P-type, it's called Zintal 4111. On the N-type, is a composite of lanthanum telluride. But still, there's a lot to understand and to, basically to understand in terms of dynamics of electrons and phonons transport in order to improve further the efficiencies and hit the goal set by NASA of reaching efficiencies beyond 20% in 2020. I'd like to thank the Thermal Energy Conversion Technology Group at JPL, the Power and Sensor System section, and if you want to have further information about RTGs, you can ask me later, or you can visit the rps.nasa.gov website or jpl.nasa.gov. And with this nice selfie of curiosity, I'll give you maybe time for one question. Thank you.
Hi, everyone. I'm a population geneticist, and today I'm going to talk to you about a really interesting story that happened in our human past. So we know that all of us originated from Africa. About 200,000 years ago, some of our ancestors left Africa, entered West Eurasia, and from there we went everywhere. By the time these people left Africa, they soon realized that we're not the only people out there. Some archaic hominins got there before us and were still around. And those include the Neanderthals in Middle East and in Europe, and also the Denisovans in the Eastern world. The discovery of the Denisovans is one of the most stunning findings in the last decade. And we still don't know too much about them, because the only fossil remains we found in Siberia were highly limited in number and very fragmentized in nature. However, surprisingly, just from this small pinky bone we found in Siberia, which retrieved a high-quality genome of theirs. So from there, we learned more about these people, including that there were once probably all over Asia from north to south. They lived up until 40,000 years ago, and they're closely related to Neanderthals. So since these humans, archaic or modern, were not that different from each other, essentially, it's not hard to imagine that by the time we've met each other, something romantic may have happened, and it did. So we found evidences for interbreeding between this type of humans by finding traces of Neanderthal DNA in pretty much everyone who is not from Africa. And we also found the DNA of the Denisovans in a lot of people from Asia, from Oceania, and also in the Americas. The specific amount varies, but it could be up until 6% of the entire genome, which is quite amazing. Most of the time, for many reasons, we actually don't like this foreign DNA that entered our system. So a lot of, most usually, what happened is this DNA entered our populations, but over time they're purged out. So after so many generations, right now, we only see these bits and pieces, traces of DNA, like here and there, telling us they once existed. But occasionally, there are something from these archaic humans entered our populations, and we actually like them. They're beneficial for us. And in that case, we'll preserve this DNA, and they're still in relatively large trunks, and we can still discover them. So one of the most classic examples of this adaptive introgression actually came from Tibet. By scanning the genome and comparing them, we found that the sequence of this one gene called E+1 matched very well between Tibetans and Denisovans, but not as well between Tibetans and the rest of Chinese, with which they're actually the closest related to. So it is really interesting, and we want to know what this E+1 gene do. And the first thing to look at is, what is so unique about Tibet? Tibetan Plateau is one of the highest places on Earth. It averages about 4,000 meters above the sea level. So at this high up, the amount of oxygen we have is less than half than what we usually have. So for most people who are not from there, we'll get immediately sick. And over time, our body will produce more red blood cells to compensate this lack of oxygen. And it does help in short term, but it's not a good thing for us in long term, because more red blood cells make our blood thick, and that can trigger other cardiovascular issues. So this E+1 gene codes for a very important protein in the blood that regulates this change in oxygens. So for Tibetans, they don't necessarily need more red blood cells than us, and they're still doing great at this high altitude. So in my research, I'm really interested in this example, and I ask a few questions. The first thing I ask is, it is plausible that the Denisovans, just like us, have multiple populations. And if that's really the case, which of these populations actually introduced the E+1 gene to Tibetans? So to study that, I looked at the genome from a lot of Tibetans from different populations, and I inferred a collection of DNA segments that possibly came from something else. And then I matched the segments with the DNA of the Denisovans and also Neanderthals. So I saw that a small amount of them matched with neither, which is okay. The vast majority matched with the DNA of Neanderthals. But we also saw two independent peaks that matched with the Denisovans, possibly indicating that these came from two unique groups. And the E+1 variation in the Tibetans actually came from this group that is most alike the single individual we have from Siberia. Another question we're interested in is, if this contact really happened somewhere in the past, when did this happen? And also when the natural selection on E+1 took place. So to answer that, we know the demography of these populations roughly pretty well, and we know the divergence time between these populations. What we know is E+1 arose somewhere in the past in the Denisovan populations, and were introduced to Tibetans through one pulse of contact or gene flow. So since we don't know when the time happens, we just simulate it all. I collected a large amount of simulations with the time of this contact and also the time of selection on the E+1 gene being the variables. And I used a hidden Markov model to infer the track of DNA that came from the Denisovans and compute some summary statistics that are informative about these parameters. And under a Bayesian framework, we compared our simulations with what we actually observed in Tibet and tried to infer the times with the highest likelihood. And what we found is pretty stunning. We saw that the admixture, or the contact between the populations, happened about 30,000 years ago, which is pretty old. But the selection on this E+1 gene did not happen right away. It happened around 13,000 years ago, which is quite a significant gap between the time of admixture and selection. So if we try to put everything together, what type of story we're trying to tell here? So the story is, modern East Asians received genetic materials from at least 3 other archaic human populations, 1 Neanderthal and 2 different types of Denisovans, at least. And this E+1 gene in Tibet came from this group of humans that is the most alike Denisovans from Siberia. It's most likely this admixture did not happen actually in Tibet. It happened somewhere in the lowland East Asia. And so the selection of E+1 gene, which make these people adaptive to the high altitude, did not happen until the mass migration into the Tibetan plateau, most likely around after the last glacier maximum. So with this, I'm going to end my talk, and do I have time for the questions?
Can anyone hear me? So my name is not Abby, and I'm not talking about like diseases and stuff. Instead, I'm going to tell you a little bit about how to look at the beginning of the universe from the South Pole. So in the picture here, it's one of our telescopes called BICEP 3, and we took this picture during the sunset at the South Pole, which lasts about a week and only happens like once a year. So just to give you a little bit of background, this is the timeline of the universe. As we see that, how to use a laser pointer? Oh, great. As we see that, we know that the universe is expanding, which means that in the very beginning, it gets really small, and when gas is compressed in a small volume, it gets really hot. So, and in fact, that in the beginning of the universe, at the Big Bang, it's so hot that everything is ionized, and photon can only travel, light can only travel a few meters away before it gets bumped into an electron gets scattered. And not until about 400,000 years after the Big Bang that the universe is cool enough to about 3,000 Kelvin and allow proton and electron to combine to form neutral hydrogen. And that's the point that the first time light can freely travel throughout the universe. Oops, you didn't see that plot. And that's the light that we're trying to probe, and we call it the cosmic microwave background. So if you have eye that can look at microwave and you look at the sky, you see this extremely uniform 3 Kelvin background radiation all over the sky. And this is first discovered in the 60s. And this is the oldest light in the universe, and it's actually the cleanest source to allow us to probe the early universe. But if you stare at it long enough and really look at it carefully, you can find this tiny hot and cold spot in this extremely uniform background. that is about 1/10,000th of a degree difference. And these are the quantum fluctuation from the Big Bang and get stretched out to like this large scale that see all the stars and galaxies that we're seeing today. So by looking at this, we can really understanding how like the universe like growth and the age and geometry of the universe. So this is like the one map that I'm going to show a little bit. By taking like a Fourier transform of the map that I just showed you, you get this temperature power spectrum. And the location of the bump and the amplitude of the bump that tell you, like I said, that we know that the geometry of the universe is flat and the universe is about 14 billion years. Those two bumps here tell you the existence of dark matter. One thing that we're interested in is like what is the initial condition of the Big Bang? Like what is the energy scale and why like we get like this quantum fluctuation to get stretched like many times out. And this is the information is embedded in the first plot here, which you can see the error bar is huge, and that's not because we're really bad at our job. And this is actually we reached to a statistical limit that in fact we only have one universe to probe. So it doesn't matter how good of a telescope that we build, this is the limit that we can get. So in order to probe that, we turn to like looking at not just the temperature of the CMB, but we try to look at the polarization signal. So in the very beginning, we think that there's a very rapid expansion happen at 10 to the minus 32 like second after a Big Bang that exponentially expand the universe about 50 to 60 eFold, which we call inflation. And this rapid expansion that create very strong gravitational wave that embedded this very unique polarization signature in the microwave background polarization. So the goal of many experiments, including mine, is to try to detect this signal so that we can learn about the inflation and why the universes happen like that we see today. Turns out it's actually really hard to probe that. So in the temperature power spectrum that I showed you earlier, this is the polarization signal that we want to probe is down here. It's another 5 to 6 order of magnitude smaller. So you're talking about nanokelvin of temperature changes in the sky. So and it's extremely difficult to detect. And that's why we go all the way to the South Pole to build these telescopes to look at the signal. And you will ask, why do we want to go to the South Pole? And I can tell you that it's not because I have an excuse to work in a penguin suit, although that's actually not bad. But many of you probably didn't know that South Pole is actually at 9,000, 10,000 feet elevation, combined with the coldest, very cold temperature, is one of the best locations to observe, do astronomy, and also away from all the cell phone and human traffic and everything. Give you a very clean environment to do this kind of science. It's just horrible for my mental health. So, I'm showing a short video of one of our telescopes scanning the sky and see the southern light. I tell you a little bit about the camera that we built at JPL, the detector that we use to detect this tiny signal. That this is one of our camera detector tile. That most of them is occupied by the antenna, and at the end you have these two sensors here for every pixel. We cool these sensors down to about 200 millikelvin, so about 0.2 Kelvin above absolute zero, and you utilize the material property for a superconducting material that give you a very big resistance change for a very tiny temperature variation. And this is what we get for 6 years of the data. As you can see, as we scan deeper and deeper, you average down the noise, and you see the underlying structure of the sky. So this is 6 years in like 2 seconds. And this is what we have like today. We many different projects and collaboration. We're trying to look at this signal, and we have a parameter called R, which is the amplitude of the inflation gravitational wave background. Although like we haven't detected a signal yet, but we think we were getting close. And by constraining the upper limit of this tensor scalar ratio, we can rule out different model of inflation and allow us to understanding what maybe our understanding of the Big Bang is wrong, or like maybe there's like more complicated things happen in the early universe. So in summary, that we have a set of telescopes down at the South Pole to look at. We try to probe the primordial gravitational wave from the Big Bang, and by doing that, we try to understand what happened of the evolution of the universe and how do we get to today the stars and galaxy from the very beginning. And finally, I want to say that this is a giant group effort. Although our team is called Bicep, and none of us really are qualified for that. But yeah, I'll stop here. Can everyone hear me? So my name is not Abby, and I'm not talking about diseases and stuff. Instead, I'm going to tell you a little bit about how to look at the beginning of the universe from the South Pole. So in the picture here is one of our telescopes called Bicep 3. And we took this picture during the sunset at the South Pole, which lasts about a week and only happens once a year. So just to give you a little bit of background, this is the timeline of the universe. As we see that, how to use a laser pointer? Oh, great. As we see that, we know that the universe is expanding, which means that in the very beginning, it gets really small. And when gas is compressed in a small volume, it gets really hot. So, and in fact, that in the beginning of the universe, at the Big Bang, it's so hot that everything is ionized, and photon can only travel, light can only travel a few meters away before it gets bumped into an electron and gets scattered. And not until about 400,000 years after the Big Bang that the universe is cool enough to about 3,000 Kelvin and allow proton and electron to combine to form neutral hydrogen. And that's the point that the first time light can freely travel throughout the universe. Oops, you didn't see that plot. And that's the light that we're trying to probe, and we call it the cosmic microwave background. So if you have eye that can look at microwave and you look at the sky, you see this extremely uniform 3 Kelvin background radiation all over the sky. And this is first discovered in the 60s. And this is the oldest light in the universe, and it's actually the cleanest source to allow us to probe the early universe. But if you stare at it long enough and really look at it carefully, you can find this tiny hot and cold spot in this extremely uniform background that is about 1/10,000th of a degree difference. And these are the quantum fluctuation from the Big Bang and get stretched out to this large scale that seed all the stars and galaxies that we're seeing today. So by looking at this, we can really understanding how the universe growth and the age and geometry of the universe. So this is the one map that I'm going to show a little bit. By taking a Fourier transform of the map that I just showed you, you get this temperature power spectrum. And the location of the bump and the amplitude of the bump that tell you, that we know that the geometry of the universe is flat, and the universe is about 14 billion years. Those 2 bumps here tell you the existence of dark matter. One thing that we're interested in is what is the initial condition of the Big Bang. Like, what is the energy scale, and why like we get like this quantum fluctuation to get stretched like many times out. And this is the information is embedded in the first plot here, which you can see the error bar is huge, and that's not because we're really bad at our job. And this is actually, we reached to a statistical limit that in fact we only have one universe to probe. So it doesn't matter how good of a telescope that we build, this is the limit that we can get. So in order to probe that, we turn to like looking at not just the temperature of the CMB, but we try to look at the polarization signal. So in the very beginning, we think that there's a very rapid expansion happen at 10 to the minus 32 like second after a Big Bang that exponentially expand the universe about 50 to 60 eFold, which we call inflation. And this rapid expansion that create very strong gravitational wave that embedded this very unique polarization signature in the microwave background polarization. So the goal of many experiments, including mine, is to try to detect this signal so that we can learn about the inflation and why the universes happen like that we see today. Turns out it's actually really hard to probe that. So in the temperature power spectrum that I showed you earlier, this is the polarization signal that we want to probe is down here. Is another 5 to 6 order of magnitude smaller. So you're talking about nanokelvin of temperature changes in the sky. So and it's extremely difficult to detect. And that's why we go all the way to the South Pole to build these telescopes to look at the signal. And you will ask, why do we want to go to the South Pole? And I can tell you that it's not because I have an excuse to work in a penguin suit, although that's actually not bad. But many of you probably didn't know that South Pole is actually at 9,000, 10,000 feet elevation, combined with the coldest, very cold temperature, is one of the best locations to observe, do astronomy, and also away from all the cell phone and human traffic and everything. Give you a very clean environment to do this kind of science. Just horrible for my mental health. So I'm showing a short video of one of our telescopes scanning the sky and see the southern light. I tell you a little bit about the camera that we built at JPL, the detector that we use to detect this tiny signal. That this is one of our camera detector tile that most of them is occupied by the antenna, and at the end you have these two sensor here for every pixel. We cool these sensor down to about 200 millikelvin, so about 0.2 Kelvin above absolute zero, and you utilize the material property for a superconducting material that give you a very big resistance change for a very tiny temperature variation. And this is what we get for 6 years of the data. As you can see, as we scan deeper and deeper, you average down the noise, and you see the underlying structure of the sky. So this is 6 years in 2 seconds. And this is what we have today. We many different projects and collaboration, we're trying to look at this signal. And we have a parameter called R, which is the amplitude of the inflation gravitational wave background. Although we haven't detected a signal yet, but we think we were getting close. And by constraining the upper limit of this tensor scalar ratio, we can rule out different model of inflation and allow us to understanding what maybe our understanding of the Big Bang is wrong, or maybe there's more complicated things happen in the early universe. So in summary, that we have a set of telescopes down at the South Pole to look at. We try to probe the primordial gravitational wave. from the Big Bang. And by doing that, we try to understand what happened of the evolution of the universe and how do we get to today the stars and galaxy from the very beginning. And finally, I want to say that this is a giant group effort. Although our team is called Bicep, and none of us really are qualified for that. But yeah, I'll stop here.
All right, so I will get started now. Can everyone hear me now? Okay, great. So thank you very much, especially Howard, for switching with me. I was stuck in some traffic this morning. So my work involves the use of methods from network science to ask questions relating to food networks and nutritional outcomes and health, of course. And so to begin my talk, I wanted to give, because this is an extremely interdisciplinary audience, I wanted to give a little bit of a background or context to this, this field that I'm researching in, and it's an emerging field called nutrition data science, or food, nutrition, health, and data science continuum across multiple different domains and themes. So this extends from the micro, the molecular, chemical level, all of the compounds that compose food, to the macro level, the food supply networks and the systems that produce food and distribute food, and then everything in between, including the interactions at the community level, so the social networks and the built environment that, in which we consume food. And so these are the multiple different domains within this field of nutrition data science, and then multiple methods, which include, for example, network science, which I use, other kinds of methods from complex systems like agent-based modeling, and then any kind of data science methods, so machine learning, et cetera. And so I would argue that this is an extremely important field right now oh, shoot, I had changed the slides around a bit, but I guess the old version came up. We are at a really important time for this field right now because diseases of diet contribute they are the number one risk factor of leading to mortality in the world today across developed and developing countries, actually out far surpassing diseases from smoking and any other single risk factor, so contributing to an estimated burden of 20% of all deaths. And at the same time, we have these huge data sets and we have these methods and tools that can help us to intervene on this system. And so, again, kind of going across these different domains, we have ingredient databases and chemical databases that help us to study the different food chemicals and molecules that compose food. So actually, something that I learned getting into this, we study around 150 different nutrients that compose food, like vitamin A, vitamin E, those kind of things. There are hundreds of thousands of different chemicals that compose food and interact in very complex ways to affect our health. And so this kind of field can help us study that, and we have the data that can help and the tools that can help us to do that. Then we have sensors and we have digital tracking devices that can measure what food people are eating, how they're eating, and connect this to health outcomes. And then we have, at the macro scale again, these food trade databases that can help us map and model supply structures and study how food is produced and distributed and how it corresponds to diseases. And so actually that's where I'm going to start talking about my work. So my work starting in my PhD was around modeling the network structure of the food supply system in order to study and trace the spread of foodborne diseases. And so I got interested in this problem around 2011 when there was a large-scale outbreak of foodborne disease carried by contaminated sprouts in Germany. So some of you may have been aware of this outbreak. It sickened thousands of people. There were 54 deaths. Most of the diseases and deaths were in Germany, but there were cases across the world, even into the US, because of transport. And despite this huge burden of this outbreak, it took investigators 9 weeks to identify the source. So what I'm showing you here is the epidemiological curve, the number of illnesses by the day of illness onset. And you can see that by the time the source was suspected and the source was confirmed, the contamination had made its way through the supply chain, and everyone who would have gotten sick had gotten sick already. And so digging into this, I quickly found that one of the main reasons that this timeline is so long is because the investigation is manual. So investigators are going out and collecting records in person. They are interviewing, like I'm showing you this diagram, the top right here, they're interviewing patients, and they'll find out where they ate, and they'll go to those places and collect records and move, and this is actually a diagram from collecting records from that 2011 outbreak. And they put this together, and they hope that they find some convergence far up in the supply chain. But this all takes a lot of time. And I would argue that tracing outbreaks through the complex system of the food distribution system requires a network approach. And so right here, I'm showing you simulations of an outbreak in the US where it started at one single location, and it quickly spread through the industrialized food system across the country. And so this is the reason that we need to take this full network perspective. And so just to back up a bit, when we are tracing outbreaks, there are 3 important parts of the problem that we need to look at. And those are, first, detecting that an outbreak is happening, understanding that there are cases related to a specific strain. So we know that there's this outbreak of E. coli happening right now. And then there's identifying the food vector, so identifying that it's sprouts that are carrying this outbreak, and then locating the source in the supply chain. So my research, I've focused on these last 2 parts, identifying and locating, and I'm going to just talk you through very quickly the location part. So this is the problem, to formalize this problem. Given a network model of the food supply system, how do we identify the location source in that network? And so a stylized view of what that looks like. So let's imagine that we have this food supply network model consisting of multiple stages: production, processing, distribution, and retailing or consumption. Then when we have an outbreak, we have cases of illness that are reported and linked to the place where the person ate the contaminated food, for example, or purchased the contaminated food, for example, if that's the retailer. And then my job as the modeler is to develop a probabilistic method to attribute a risk or a probability to any of the feasible sources, the feasible nodes in this network being the true source that caused the outbreak, and then developing a probabilistic estimator for that location. And so this is, of course, a very stylized view of these kind of methods. But I actually moved to Germany to implement this methodology and work with investigators at the German equivalent of the FDA to use this methodology. And we modeled the network structure of the supply chain in Germany. And I'm not going to talk about this now. And then when using this model, we were able to, in retrospectively, identify the location source of the outbreak. It was at a farm in northern Germany, weeks earlier, and of course thousands of diseases earlier than was done in real time. Now, of course, this is going to be much better after the fact when you have all of the clean data. But this is just an illustration of how these kind of methods could help contribute to solving the investigation problem. Now, in my last minute, I want to actually bring back to a lot of this research was what I did during my PhD and first postdoc I did in Germany. After this, I still think the problem of foodborne diseases is really important, but the really trillion-dollar question is diseases related to diet and nutrition. And I started the talk by mentioning this huge burden of disease from diseases of diet. So I was thinking, really, in my career going forward, how can I use these methods that I've developed, these methods from network modeling, models of the food supply system, to ask questions of nutrition? And so for my postdoc at USC that I've just started last month, I'm doing a very big data analysis to study food systems and consumption patterns across large populations. So we have a smartphone data set of 700,000 individuals in the LA area, and we have their micro patterns, we have their mobility, geolocation every 5 minutes, and we're going to be studying people's activity patterns related to their consumption, or what we're calling food foraging behavior, and seeing how this translates to health outcomes. And so stay tuned for some postdoc work, but yeah, glad to be here in this very interdisciplinary group, and good to talk with a lot of you. So thanks.
Okay, my name is Frances, and I'm actually a microbiologist that lives in the physics department, so it's been a fun journey for me. So I'm going to talk to you about these ideas that we've been looking at about naturally produced antimicrobial peptides or antibiotics, which are actually secondary metabolites, and really their natural role, or what we hope or are interested in, their natural role in actual biological communities. So in general, our group studies biophysics of natural microbial networks. So that just means we're interested in various amounts of bacterial communities living in your gut, which is very popular right now. Everyone's very interested in that. The ocean or the soil. And so these different environments are made up of a whole host of species of bacteria. And so we're interested in how they make choices with each other, how they send chemical signals, and decide how to seed where they are and make community. So I particularly, in this path of communication between interspecies communication, am interested in, we are interested in extracellular vesicles, which you can see here are released, these are TEM images, where the bacteria releases these vesicles through its normal life. And all bacteria that's been studied has shown to actually produce these. And so what do these extracellular vesicles do? So they look like these mini cells, as you can see here. And through its life, they release a handful of them, and they package them with all types of fun biomolecules. Things like proteins, signaling molecules, like PQS. They also act as decoys for immune responses, so they can protect the bacteria. They've also been shown to actually elicit immune response in humans. And more interestingly for us, we've been interested in nucleic acids, DNA, RNA, things that have been packaged in these vesicles. And so why are we interested in the DNA? We're actually interested in horizontal gene transfer. So horizontal gene transfer is an amazing tool that bacteria use for biodiversity and fast adaptation. This is the ability for them to just pass DNA back and forth instead of having to reproduce. And so that leads to fast acquisition of genes that maybe lead to antibiotic resistance or pathogeneity. So the idea of pathogeneity is a really transient state. It's not a constant state always for different pathogens that you know of. And so listed here are 3 different forms of horizontal gene transfer that microbiologists have been studying for years. And we've introduced a 4th mechanism mediated by these extracellular vesicles that we study. So this is the model, and a couple of our early papers have shown that this transfer is done in interspecies. But in general, just to get an idea of what's happening, is you have your donor cell, your bacterial cell, and it's growing. And at some point, it releases a bunch of vesicles, some packaged with some type of DNA that can be tracked, like an antibiotic resistance. And in some space and time, another cell will receive it and acquire that trait, and we can track that trait. So reversely, we're also interested in these secondary metabolites like I talked about. So antibiotics and antimicrobial peptides, we have used as humans for a long time in really large quantities for our own health. But they're actually, like I mentioned, naturally produced in the environment. And so there must be an actual natural use for them in these bacterial communities. And so we're interested in what they're used for. So secondary metabolites have been shown to do a lot of things, regulate genes in the cell itself, in neighboring cells, interact with proteins, and more importantly, signaling between cells in the community. So we've taken this idea of naturally produced secondary metabolites and the release of them into the environment and asked why would they do this. And so if you think of an environment when bacteria is stressed out or needs something, we imagine that it might release all of these molecules into the environment and ask their neighbors to give them traits that they might need to adapt. We call this a gene stealing model. So as you can see here, one cell will start to release molecules into the environment because it needs something. And some neighboring cell, these molecules will interact with the membrane and induce curvature to that membrane, eliciting vesicle formation, increased vesicle formation, thereby increasing the DNA that's packaged in these. And then the cell that released these can actually acquire this trait faster. So these are adaptive traits that are happening as the cells move in time. And so the way that we've studied this, and we've shown that in vitro, if you add lots of vesicles to cells, you'll get faster uptake rates, faster adaptation, and gene trait acquisition. We've also created an engineered model where you can tether the trait into a vesicle, deliver them, and lead to the trait that you want. And that's been patented. But this one is more interesting to us because we're really interested in these environments. So we just have engineered different bacteria so that we can track them by color. And we've added them together and then expressed these exogenous molecules, particularly ones that interact with the cell membrane, and tracked that acquisition of antibiotic resistance over time. That's the wrong direction. And so what I've shown here is we have three different, two antibiotics and polymyxin is an antimicrobial peptide. And all three of these interact with the cell membrane and induce curvature. And so as you can see here, when that is introduced into the environment, you reduce the time to trait acquisition nearly in half. So broadly, what this really begs us to ask what's happening in the environment and what is curious just naturally for environmental reasons, but really for human health, is how is this vesicle-mediated gene stealing affecting adaptation and biodiversity in bacterial communities? It's a thing that we haven't known that was existing for a really long time, but clearly has. And what does that mean for what is happening? And how do we, as individuals, as we use antibiotics and antimicrobial peptides in the environment, how are we eliciting this gene stealing to happen in an unnatural rate? So that's what I have for you today, and I'll thank my lab and take any questions.
Hello, everyone. First off, I have the pages as a prop, but also because I want to thank everyone for being here today, and also the organizers of this very thought-provoking conference. Now, I have to say that I feel no pressure whatsoever being one of the few people from the social sciences / humanities in here presenting. Actually, I'm a bit nervous, so that's why the prop. But I'm here to tell you that, about my work and how I think that my work, along with the work that all of us are doing here, are part of this larger conversation, which is the mission to create useful knowledge. These are my my research interests: Southern California, 20th century, 21st century as well. Transnational youth cultures, how youth cultures across time and space and borders exchange music, literature, and also social movements across borders. And this is, I think, what brings us together, which is how we produce knowledge with the firm intent to create better conditions for our neighbors, communities, and also our fellow humans. So I'll be talking a little bit about my work, but also how I understand that I create this useful knowledge that is shared. So as a historian of the late 20th century, Southern California, I studied the change over time. And the connections between and among Latinos, Latinas, Latinx in Southern California and Latin America throughout, as I said, cultural productions. In my work, in the dissertation work, it's more about the late 20th century, but also to elucidate, to make visible that is part of this longer history that connects LA with Latin America. In particular, I examine a music genre called rock en español, or the LA variant, which is rock angelino. And why the music genre, and also why this moment? Well, we can think of this as part of these youth cultures that have been circulated and created in Southern California, and how they offer us a more complete understanding of how cultural expressions in the 1990s became a strategic, symbolic, and spatial manifestations of the presence of Latinos, Latinas, Latinx in Southern California, but also the solidarity and visibility for urban Latinos that go beyond our understanding of Latinx as mere laborers, immigrants, or just social activists. And also by centering the cultural productions generated and circulated by Latino immigrants and also US-born Latinx in Southern California and Latin America, my work offers that new point of intervention for our current historical research on the near present and the late 20th century. These were decades of when a multitude of cities in Southern California were transformed by the industrialization, white flight, and also the arrival of different communities such as Asian Americans and Latinos who by the 1980s, in the case of Latinx residents, have become the demographic majority in these municipalities and how they transformed the urban space of Southern California. So here we see a Mexican teen mall in the city of Linwood, which is not far from us, or an all-American street in the city of Huntington Park, and how it has become a primarily Latinx commercial district. So my study also or helps us understand this critical period of the late 20th century, a period where Los Angeles experienced an urban insurrection in April 1992, which we know as the LA riots, and also how the California electorate at the time approved an anti-immigrant proposition such as Proposition 187, did away with affirmative action in state institutions as it was repealed with the passage of Prop 209 and Prop 227 that was also approved by the California voters, which eliminated most public education in public schools in California. So how did the youth at this time responded to this marginalization? I look at what I define as ephemeral forms, which are makeshift spaces that Latinx youth created mostly in and around Latinx neighborhoods in central Los Angeles, and for them as a way to create their own spaces and to make spatial claims in the American metropolis. This through the exchange of cultural production and also political advocacy that promoted a more democratic societies in Latin America, and also perhaps they want to just listen to a show, but also to create this kind of political awareness and activism to create more equal and just society in the United States. And in here we see not only that it had that function, but also how the participants of this created and mapped out the American metropolis by adding maps into these flyers for a music show. But in here, we can also see how, by doing this, they also respatialized and re-understood the space around them in these communities. So while most of the current historical scholarship has correctly focused on the riots, on the state xenophobia at the time, and also even gang violence, the growth of communities of color, and in particular Latino immigrant cultural production, has been under-examined. I argue that by analyzing the thriving cultural productions by immigrants and second-generation youth, we're able to understand how people who are often placed at the margins have made their presence bold and visible in the state of, in a period of xenophobia that happens in the 90s, and we can continue to think about it in the present moment. Furthermore, the examination of cultural expressions as historical archives made and shared by immigrants and people of color also enables us to better understand the multiplicity of voices that have narrated and intervened in the changes that Southern California metropolis witnessed at this time. So in some of this, I do this obviously in my work in academia, but also I go beyond this. I do this through Instagram, actually. It's as a public scholarship project in which I crowdsource different archives of sources that flyers, concert photos, tickets, and other ephemera, along with captions that connect the photo to the larger historical period. This type of public scholarship allows us to examine and visualize how the creators of these flyers and also these independent publications, who were either immigrants or later generation Latinx, mapped out and made a sense of their presence in the Los Angeles metropolis, both cartographically and also sonically. And this brings us to the last, to me, what I understand as the useful part of my work that I offer. The sharing of cultural expressions that emanated from Latin America to counter dictatorships in the case of Chile and Argentina, to do away with vestiges of fascism in the case of Spain, and also to advocate for more democratic aperture in the case of Mexico, informed the way in which Latinx youth articulated their responses against xenophobia directed at them during this explosive period in the 1990s. But also how they have continued to make expansive claims for citizenship and belonging in US society and in multiple societies. These are the two examples that I look in my work, how transnational networks of communication developed by perhaps music fans or youth inform the way how youth and community members have engaged in political participation practices that go beyond electoral politics to push back against structural racism, demand environmental justice in the case of Southeast Los Angeles, which are one of the most polluted communities in Southern California, and in the quest to gain environmental justice, in addition to this persistent advocacy and defense of immigrant rights at the local, regional, and national levels. The sharing of this useful knowledge and of the past also empowers communities to recognize the historical presence in the American metropolis, while also understanding their role in advocating for a more just metropolis and more inclusive and democratic nations and societies. Thank you for your time.
Okay, thank you for the chance to be here today. I'm here at UCLA. In 1953, a man now known to the medical literature as HM began a journey that would one day make him the most famous case study in neuroscience. Hoping to find relief from uncontrolled epilepsy, he underwent an experimental procedure, the surgical removal of his medial temporal lobes, which is a brain region that at the time, its function was uncharacterized. Although the surgery proved ultimately successful in eliminating his seizures, his doctors were dismayed to discover it had an additional side effect. He permanently lost the ability to form any new memory for any detail of his life. This single case study launched an entire field of research into the nature of memory and the role of the medial temporal lobe in memory function. We now know a lot about the biology of this region. We know that it's the first to experience cell death in Alzheimer's disease, and that the corresponding memory loss is likely due to the fact that the hippocampus and entorhinal cortex sit at the top of a long information processing hierarchy, whereby information about the world comes in through our senses, goes through many stages of processing, and eventually arrives here, where these two regions, the entorhinal cortex and the hippocampus, play a critical role in translating that experience into a memory. And yet, although we know all of this biology and these circuits, we don't have a clear picture of how the human brain does memory. We don't know the neural codes for laying it down or retrieving them again. But more importantly, for the countless people around the world who suffer from conditions that cause memory impairment, we're asking what goes wrong in these processes and are there ways to fix them? So more than 60 years after HM had his medial temporal lobes removed, tissue resection remains a state-of-the-art treatment for people whose seizures don't respond to medication. Prior to resection, however, patients will undergo extensive testing to determine their precise seizure onset zone so as not to lose too much brain tissue. After using non-invasive techniques to identify candidate epileptogenic regions, a neurosurgeon will implant electrodes into the suspicious regions. And these electrodes can pick up the brain activity around them and thereby trace out the electrical signature of a seizure. So patients will typically have these electrodes implanted and then remain in the hospital for 1 to 2 weeks until enough seizures have been captured that they can have a concrete treatment plan. Now for me, as someone who is interested in understanding how the brain does memory, these electrodes provide a really unique and special opportunity because most of the time that the patients are in the hospital, they're not actually having seizures. and their brain is just doing normal brain things. And so most of the patients who come through our program agree to allow us to observe their brain activity while they take some tests or play some games. And then we can use that information to learn more about how their brains are accomplishing the tasks we ask them to accomplish. I'm especially interested in recording from the hippocampus, of course, because it is the pinnacle of the memory processing hierarchy. And if we had more time, I would love to tell you about all the many, many fascinating things we have learned from just listening in on the brain about how the brain does things. But today, I'm instead going to focus on something that might be even more impressive, and that's what we've accomplished by talking back to the brain. So in addition to reading out the electrical activity in our brain, our participants' electrodes can also send small electrical pulses back into the brain. And we asked whether we could use this electrical stimulation to give the hippocampus a boost when it's getting ready to learn something. To test this, we showed our participants photos of people they'd never seen before. And for half of them, right before we showed the photo, we sent in brief, tiny pulses of electricity into their entorhinal cortex, which we thought would prepare the hippocampus for learning without disrupting its own processing. And then later, we showed the patients photos again of either the same people or people who looked similar to them and asked if they'd seen them before. And our hypothesis was that they would do better on the items that had received stimulation than the items that didn't. And sometimes it worked. So here you have the data of one patient who, in the stimulated condition, got about 25% more answers correct than in the non-stimulated condition. So that's very exciting. But other times, stimulation didn't really seem to do very much. And when we looked at all of our data more closely, we saw that whether or not stimulation had a positive effect on memory was really dependent on two critical factors. First, if you split it up by whether we stimulated in the left or the right hemisphere, you can see a pretty distinct difference that we can summarize here by the mean change in how many they remembered. And you can see that on average, people do about 10% better on the stimulated trials in the right hemisphere and it doesn't have much of an effect in the left hemisphere. We also wanted to look at whether we applied stimulation either in the entorhinal cortex proper, so in the gray matter of the entorhinal cortex, or in the white matter pathway that connects the entorhinal cortex to the hippocampus. And when we broke down the data further into that, we found that, again, the right was better than the left, but also white matter is substantially better than gray matter, such that we got a pretty good memory enhancement effect from white matter stimulation. And that wasn't true just in this person recognition task. We also did an object recognition task where we found a very similar pattern, a face name association task where we found a similar pattern. And in this verbal memory task, even though they're pretty similar, we found that white matter was the most consistently effective. So the big message of our research so far is that direct electrical brain stimulation does have the potential to enhance human memory, and it's really exciting, but it also matters a lot where and probably when you stimulate. So there's a lot of work left to be done to identify the best parameters and the best practices to really find the exact way in which stimulation is going to have the most enhancing effects for memory. But given the sort of global health burden of various conditions that cause memory impairment and just the general devastation that this impairment causes, and the fact that there are currently not really any truly well-functioning treatments for memory enhancement, it's exciting to be in this field developing a technique that shows some promise and that we hope will one day change the lives of people with memory disorders. Until we can replace our brains with memory sticks, we're gonna try. So thank you for your attention.
All right, so hi everybody. My name is Jasmine Plows, and I'm originally from New Zealand, but about a year ago I moved here to Los Angeles to do my postdoc in the lab of Dr. Michael Gorin. First at USC, and a few months ago we transitioned to Children's Hospital Los Angeles. And today I'm going to be talking about my research, which looks into why intervention before birth could be the key to curing the obesity epidemic. And I thought I'd start by telling you the story of how I myself got involved in this research. And that story starts when I was just 15 years old, because that is when I met my now wife, Mary. So Mary and I met in high school, and we became friends and eventually started dating. And over the course of our friendship and our relationship, I noticed that Mary suffered from more than her fair share of health issues. So nothing too big and serious, but for example, she has a common metabolic disorder called polycystic ovarian syndrome. She suffers from chronic migraine headaches. And when she was just 20, she required a fairly emergency surgery to remove her thyroid gland after she developed an autoimmune disease. But at the time, I just chalked this down to simple bad luck. Until one day, I was attending a lecture as part of my studies, and my ears began to prick up, because what this lecture was telling me reminded me a lot of Mary. You see, I knew Mary had been born as a result of complications. She'd often mentioned offhand that she'd been born very small, that she'd almost died when she was born. But when she said that she was delivered at 37 weeks gestation, I didn't feel like that sounded very premature until I went to this lecture. And that night, I went around to her parents' house for dinner, and I asked them if I could see her medical records from when she was born. And they kindly went and fished that out for me, and I have a copy of a doctor's note here. And sure enough, exactly as I had suspected, based on that lecture I attended, were these three words: asymmetrical growth retardation. And that's a picture of Mary with her grandmother. She was born under 3 pounds. And when I read these words, it was like a little light bulb went off in my head, and I knew that this was what I wanted to focus on in my research. So what is asymmetrical growth retardation, and why does it matter? Well, these days we'd more commonly refer to this simply as fetal growth restriction, and it means exactly what it sounds like. It means when a baby does not grow properly in the womb. And there's many causes of this many mechanisms through which this can happen, but the most common is a problem with the placenta. So in normal pregnancy, when a mother eats food, that food will pass through the placenta, through the umbilical cord to the baby, and cause the baby to grow. But in fetal growth restriction, often there's a problem with the placenta, which means that even if the mother eats all the right things, that food will not adequately reach the fetus, and the fetus will not grow properly. And indeed, that's what happened in the case of Mary, and in around about 10% of pregnancies in the US today. But why does this matter? Well, it matters because it turns out that these small babies are at increased risk of obesity, type 2 diabetes, and heart disease, 3 of the biggest health issues facing this country and the world today. But why is this happening? Well, scientists hypothesized over this for a long time until they came up with this theory. And it's that in the womb, the fetus very cleverly adapts to the environment that it experiences. So when a fetus is underfed in the womb, it assumes that it's going to be born into a world in which it will be underfed. And so through presumably epigenetic mechanisms or changes to the expression of genes, it will do things like turn on genes involved in storing fat and turn off genes involved in things like needlessly burning energy at rest. And you can imagine that even 200 years ago, this would have been enormously advantageous, because chances are that these small babies would have been born into a world in which there was hardly any food. The problem is when these babies are instead born into the current obesogenic environment that we see today. And it is this mismatch between the in utero environment and the external environment that's thought to contribute to disease. Okay, so that's the small babies. What about big babies? What about babies like this one, born just last month in New York City at 15 pounds and 15 ounces? Are these babies better off by that logic? Well, not exactly. It turns out that just like the small babies, big babies are similarly at increased risk of obesity, type 2 diabetes, and heart disease. And the reason this is because the majority of large babies are born as a result of a pregnancy complication called gestational diabetes. And gestational diabetes simply means when a mother has too high blood sugar during pregnancy. And again, this is really common. It happens in at least 15% of pregnancies in the United States. The problem with this is that the high levels of sugar in the mother's blood easily flow through the placenta to the baby, causing the baby to essentially just be munching on sugar for the 9 months before they are even born. And so these babies are essentially already born obese. These big babies tend to stay big the rest of their lives. We all know it's difficult to lose weight once we already put it on. So what we end up with is this really interesting U-shaped relationship whereby we have small babies, fetal growth restricted babies, at high risk of disease, and then as birth weight increases, it decreases till we get this optimal level here, and then metabolic disease risk increases again at this side of the spectrum. So what this is telling me is that unless everything goes completely right for you as a fetus in the womb, unless your placenta is perfectly formed and your mother has the exact right amount of sugar in her blood, chances are that each and every one of us lies on one end of this curve or the other. It's a spectrum. It's not just two different sides. And so to me, this indicates that a large amount of the variation that we see in our individual risk for obesity and diabetes and heart disease is to do with what happened to us before we were even born. And you know, that's not even considering things like genetics, which we also know contribute. So that's where my research comes in. I'm trying to figure out which factors might contribute to these suboptimal conditions in the womb. And of course, if we better understood which factors contribute, then we could potentially design interventions. So right now, I'm working on a clinical study which is called the Sweet Pea Study. It stands for Sweeteners in Pregnancy Effects on Adiposity. And I'm recruiting women in the early stages of pregnancy and looking at their artificial sweetener consumption and looking at how their fetus grows and develops with that artificial sweetener consumption. The reason why is because back home in New Zealand, I did studies in animals where I found that artificial sweeteners contributed to suboptimal conditions in the womb in mice. So if you or anyone you know is in the early stages of pregnancy, please visit our website, sweetpeastudy.com, to see if you might be eligible. So in summary, both small growth-restricted babies and large gestational diabetes babies are at increased risk of later metabolic disease. However, the contributing factors are poorly understood. And if we better understood these factors, then we could maybe develop new treatments. You can imagine that would be very exciting because then we could prevent some of these huge illnesses that are really affecting our society even before birth. So thank you, and I invite you to ask any questions.
Thank you for the invitation and the presentation. Let's check that everything works. Can you hear me in the back? Okay. So in this talk, I would like to tell you about a situation which is pretty unusual in science, physics being applied in math. As you surely know, usually mathematics is being applied in physics. For example, differential geometry, which is a branch of mathematics that studies curved shapes, is a crucial tool in Einstein's theory of gravity. In our case, physics is applied in mathematics. And in particular, string theory turns out to be a very useful tool in the knot theory. Okay, but let's hold on for a minute and ask a very basic question. What is a mathematical knot? So, in general, it's something similar to what you willingly do at your boat trip or unwillingly do with your computer wires, but there is one crucial difference. So in mathematics, when we tie the knot, let's try with the simplest non-trivial one, we always glue the ends. And then we forbid any cutting or gluing. We allow only for continuous transformations, like anything like that, and then it works. Okay, but why we impose all these rules? We do it because then, if we follow them, the knot, whatever we do continuously, always stay the same. Okay, so now we are ready for the main question of this talk, why physics is so good in describing knots. In order to understand it, let me propose an analogy. Imagine that we would like to say something about the whole globe basing on LA. So, looking around, we might think that the Earth is flat and full of traffic jams, which is not the case. However, if we are smarter, we might think that there is a lot of salty water around the world. So as you see, it's not that easy to determine global properties basing on the local ones. And it turns out that physics is very good at it. For example, starting from just moving particles, moving molecules, we can describe the whole atmosphere. Or, starting from the rules of continuity, we can describe a knot. Let's look at examples. So, before physics entered the game, knots were described as flat objects like this with a couple of crossings. People just counted these crossings. Of course, they were smart people and they did it pretty well, but there was a kind of problem because the knot, as you see here, it's a three-dimensional object in three-dimensional space as we see it. However, this description was totally two-dimensional, totally flat. And Edward Wheaton, a physicist, introduced the first three-dimensional description of the knot, which was based on the idea of following how the physical field, which is a little bit like electric or magnetic one, you can imagine it as just these red arrows which are attached to the knot. So he started thinking how this field changes when we go around the knot. Turned out to be very fruitful, and in 1990 he got the Fields Medal, the most important award in mathematics. The next example is much less impressive but more local. So in order to understand it, let us look at this picture. On the left-hand side, you have a knot, this flat picture with crossings. On the right-hand side, there is something new and strange. It's called quiver. Quiver is just something made of a couple of nodes, potentially a lot, connected by arrows. Despite this simple definition, there is a whole branch of mathematics studying these objects. It's called quiver representation theory. Okay, but you may ask, why this strange object is here in this talk about knots and physics? And you wouldn't be alone. For years, knot theory and quiver representation theory were developed totally independent. However, together with my collaborators, we showed that using physics, we can see that these two very different objects, two very different disciplines, are very closely connected. So let me summarize with two main messages. First is that physics is very good in describing knots. The second is the reason behind it. This reason is the ability to translate between local and global properties. Thank you.
Hi. Today I'm going to talk a little bit about fear and anxiety. So first of all, the only background you need to know is that mice do not like swimming that much. So this is the experiments. You can follow me and see how we actually perform experiments. So this is the fish tank. The fish tank, so filled with water, and the left side is a foam board. The animal, when we put the animal into the fish tank, the animal can swim around, but it doesn't really like that. So you can climb on the safe island and stay there and grooming and cleaning itself. So we can see for a normal animal, when we put the animal in the water, it can kind of struggle and swimming around and climb on the safe island and stay there, explore a little bit, and then start grooming and stay there for as long as he like. Then when so we can see that animal just kind of have some selective feelings about this side. It's kind of like a drawing thing. So there is no threat on this safe island, but if we manipulate part of the brain, it's actually a small portion of the hypothalamus. So let's see what's going on. The technique we're using is called optogenetics. So basically, in brief, it's like using virus tools to express ion channels on the cell membrane. Then we can use LED light to drive the activity of the neurons in the brain. So we can increase or decrease the activities. Here we just activate part of the brain and see what's going to happen. So the animal, whenever the animal climb on this safe island, we start the light stimulation So the animal just feeling something really bad going on and then jump back into the water. So the most straightforward conclusion we can reach here is that what the animal feels on this side when this small portion of the brain being activated is even worse than feeling when it's swimming in the water. So we can see that this is like artificially generated negative feelings. So what is really useful? So we can see in the reality in nature, we definitely have kind of like fearful feelings, especially when you're chased by a bunch of snakes. So this is the BBC documentary second season. The lizard was chased by a bunch of like dozens of snakes and being catched. Finally, it escaped. But we can see that this lizard can perceive this kind of visual movement of snakes and really can sense the danger is approaching. So this kind of negative feelings can protect us from even, because if you want to image in the reflex system, you have to be touched by someone, then you can give a response. But when you're facing something really dangerous, you need to develop a risk assessment and have a prediction and do something before that happens to save your life. So about how to really understand what's really going on in the brain, most of the time we start from something really simple. This is the hydra. This is the most simple structure that can have a nervous system, and it can perform a locomotor behavior as well as a feeding behavior. But when we're trying to probe the neural circuits in there, we find if it's a single neuron, it's a unicellular organism, it directly receives activation and do something in response. And then if you have 3 cells, you can have an input cell, and you can have a relay cell, and have an output cell. And then by the time you're getting more and more, you have a more complicated interpretation of the signal. But even for hydra that may seem of a very simple organism, you can have a very complicated neural network. Seems impossible to really see what's really going on in this neural network. So from an evolutionary perspective, our human now have the biggest, largest number of the cortical neurons. We have like 86 billion neurons in the brain. And so the way to probe the functionality in the brain is really complicated. So we're still trying to simplify this problem by mimic, because we have some developmental similarity. This is a human. We may have some development similarity with rabbit, cow, or pig. So most of the time, we're just using mouse models. So from mouse models, there is a lot of work behind the video. So I'm going to simplify this interpretation. So for the anxiety model, basically, we firstly have a detection of the signals, like what's really going on in the outside world. And then our part of the brain can interpret the signal and the information and process what's really going on. And like a reward system can have an evaluation, like whether it's positive, it's negative, it's dangerous or safe. And then we initiate the responses here. So why these experiments could be really useful? Because what we find is when we're silencing this part of the brain, we find that some people, some animal may be sensitive to some stimulation and develop kind of like an anxiety disorder. But when we silence this part of the brain, the animal kind of like calmed down. So could be a potentially a very promising therapeutic target for disease treatment. So that's it, and we'll be willing to take questions.
Can everyone hear me? Cool. Yeah, so my name is Riley Connors. Sweet, thank you. I'm a postdoc at Caltech. I've been there for about 1.5 years. At this building, which kind of looks strangely pieced together. And I'm going to talk to you about black holes and why they're so bright. So why it is that they're able to convert huge amounts of gravitational energy into light. And just for some perspective on that, so this is obviously the sun. And like a lot of other stars, it's an extremely bright object. Something like 10 to the 25 household light bulbs in terms of how luminous it is. But it's not for you, not entirely unsurprising. It's not so difficult for your mind to comprehend because it's huge, right? I mean, you can fit 1 million Earths inside this object. So that's not entirely surprising. But if you take a black hole and say, this is an example, it's obviously an artist's impression, it's not a real image, right? But this is what you get if you have a binary system of 2 stars, and one of those stars is heavy enough so that when it dies, the particles can't counteract the gravity, and this thing just crushes into a single object, like an infinitely dense object. We call that a black hole, right? Because there's a point at which light cannot escape, so it's black. If that gets close enough to a star, it decides that this is good food. The tidal forces that are exactly the same as the moon's effect on the Earth that cause the tides, right, cause gas to be stripped away from the star. And this eventually, due to different effects, but mostly viscous shear forces between layers of gas, this gas will eventually move closer and closer into the black hole, fall in, and huge amounts of energy are outputted from that in various forms. And just for some perspective on why that's kind of shocking, this black hole is something like the size of downtown LA, right? So you've got something that's as heavy as a star that's crushed into a few kilometers in radius. And that's exactly why this kind of object is so efficient at taking huge amounts of energy and then churning it back out again. So dead stars are small, but they're very vicious objects. But the interesting question for me in the research that I do is, how do you transform that gravity into light, into the things that we observe? What's the physics that goes on there? I'm sure most of you in the room saw this image when it came out back in roughly April, I think. This is a huge collaboration of many different radio telescopes that join together to try and actually capture the image of a black hole shadow. What you're looking at there is not necessarily the black hole, it's just the effect of how the black hole bends light around itself, and that creates what's called a shadow, right? So the light that you see at the top is really probably coming, well, it definitely is coming from gas that's actually behind the black hole. But the black hole's gravity is so strong, it bends light around it, right? And then this would be from gas that you see on the front side. But there are lots of elements there. There are lots of different physical processes happening. By and large, you're just converting gravitational energy into light, but a lot of things happen to the gas along the way. Some of that gas just falls into the black hole and never really emits a lot of light. Some of it gets launched straight back out in these jets. We call them relativistic jets because the gas moves at relativistic speeds away from the black hole. So there are certain times during a black hole's life when it becomes an extremely efficient recycler, right? It converts that gravitational energy very well into kinetic energy, and then sometimes that energy may never go into light. It may never fall into the black hole, but instead it gets fed straight back out. So if you have something like this object here, which is a supermassive black hole at the center of a galaxy, this kind of object has a huge impact on its environment. So then you start wanting to answer questions like, what kind of effect do black holes have on why the universe looks the way it looks, if so much energy can be transferred and then sent straight back out? I'm not going to go in, you don't need to read all this stuff. It's just, I put this here because it's a really nice image. So my research is basically just to take data that we have from satellites, mostly spectral information, so I look mostly at X-ray spectra, and by modeling that, try and understand how the gas that approaches the black hole behaves, what's the physics going on that causes these jets to get launched out, that causes all kinds of different kinds of emission. We observe these things all the way across the electromagnetic spectrum. They're quite bright, way across, from radio to X-ray, gamma rays. But there are a number of open questions that don't necessarily apply directly to what I'm doing right now, but are the biggest questions that we have when it comes to black holes. One of them that I've already mentioned is, what's the impact of black holes on the universe? This is an actual radio image of a galaxy. And you see that the brightest regions of this galaxy are actually very far away from the galaxy. And this is the galaxy here, and this is because The black hole is transferring such a huge amount of energy out into its environment that it's heated up some of this gas around here, and this happens to be extremely bright in radio emission. You know. Another open question is, how did black holes get so big in the first place? So supermassive black holes can range from something like 1 million times the mass of the sun to tens of billions times the mass of the sun, right? How did they get that big? There's a very simple calculation you can do. You say, I'm going to take gas that falls into this black hole at a certain rate, and then say, how big should it be, given that it started from this point in the universe? And you basically can't explain these huge masses that go up to like tens of the billions of solar masses. So there are all kinds of astronomers out there who are trying to figure out, mostly with simulations, but also with observations, of course, how you can merge these galaxies together and combine these black holes to get huge masses. And even that doesn't seem to be a very satisfying explanation. So it's a very big open question. Is Einstein's theory correct? This is a very typical topic of research when it comes to black holes and other very compact stars. We try to use whatever observational method possible. You know, this is just a little cartoon example of black holes merging together, which is what LIGO does, the Gravitational Wave Observatory. And this is a good example. They were able to actually measure the mass of that black hole just because of that image. And this just comes from modeling, you know, what do we expect the space-time of this black hole as predicted by, or as a consequence of Einstein's theory. What should it look like? And then what should our images look like? What should our gravitational wave signals look like? What should all these things look like? And another one, which is a slightly more, it's something I work on a little bit, but it's maybe a little more of an aside, is, you know, we see these supermassive black holes. We're not entirely sure how they formed. We see black holes in our own galaxy. There are something like between, I don't know the exact number, but it's dozens that we've observed. There are obviously many more. And we know they are the result of dead stars. But we also wonder, could there have been black holes that formed at the very beginning of the universe? We had a talk earlier today about this inflation theory. There are ideas out there that black holes kind of like sprung up during inflation, and then they've been growing ever since. And that's another idea that is actually extremely difficult to test, but is something we're doing. I think I'm done.
So my content area is mathematics education. In particular, I'm interested in the learning science and how the students get acquired some of the mathematics concept and feeling like a mathematics concept. And teachers' education in secondary and middle school education. This project was on the agenda of University of California, Davis, when I was in my last round of the postdoc researcher funded by National Science Foundation The full title, Formative Assessment on Mathematics, Current Status and Guideline for Future Development, when I'm still focused on my quantitative research methodology and how to apply this with fancy and complex modeling to modeling the learning science data and figure out the factors, particularly on the pedagogical level, the factors going to impact and navigate the students' learning outcomes. So why do we need the formative assessment? We already have so many summative, what we call standard tests across the whole K-12 education, CST, Spark, because first, United States doesn't do so good work internationally compared with the top ranking of the education students' performance. So we try to catch up with them. And the second, because the summative assessment, which we usually use, doesn't work doing so much work. Even though a lot of products like with Johnson and Stark try to navigate it and progressing to change the status of students' levels and their performance scores, but because of the features and because of the demographic information, in particular in California, are so diverse, we have a lot of subgroups, English language learners, underrepresented, and minority. They are still challenging and struggling with some basic and fundamentally skills and concepts. They cannot really progress as we want. So introduced with formative assessment, it's pretty brand new, it's flexible, and integrated with the whole classroom instruction. It's not separated like assessment, it's much more like instructional based, it's much more like contingent and catering to the students of each levels and different in different settings. So the second part of the graph, which I put it here, is the major content, Common Core State Standards 8EE, what we will discuss today. The title is expression equation for the linear expression equation graphical proportional linear. That is a term, but in particular used for 8th and 9th graders, used to evaluate the students' mathematics content level. So that is how the summative assessment is going to tell us. That's a matrix, a lot of levels. We can see a lot of red flag. And usually the accountability system of the school principals and administration, they are looking at this map and decided where the student learning. But from the summative assessment, we cannot hear what the students get, what the students missing, and why do they fail, or why do they success. However, we think formative assessment actually can do this work. Look at the eye clickers. The teachers just use them to point to each cycle of the classroom. They can point out and map where the students are missing, why are they disengaged, and why didn't they get this episode. And also the teachers use the individual whiteboard. It's quite assignment based. So the teachers can check out their understanding and ask their questions and address the misconceptions just in the classroom, but not in this specific anxious test situation. And also formative assessment have been identified numerous times by the research-based evidence. I will not read through, just mention some very typically data that I'm very sensitive with. First, from the last line of formative assessment meta-analysis, numerous studies from 180 of the same empirical study used formative assessment intervention across different levels from K-12 preschool to high school, and different domains from music, engineering, mathematics, or even agriculture. All of them proved they have a very strong effect, which is to say it's not only they are so effective to promote your learning, but the effect size is pretty powerful. It's almost mediocre. This might not be rare in the STEM in other field, but in social science with such effect size, which to say our program are pretty successful in the last decades of use formative assessment. So that is my research question. I will not read through, but we will show you the whole research design to see how we will do the research to prove this actually work. So the three-dimensional classroom assessment is taken from some literatures. We taken from some literatures and in particularly to realize three purposes. First, to make the formative assessment in compliance and fully alignment with the Common Core State Standards 8EE, which I showed you just now. And second, formative assessment should challenge the students the way cognitively. So they will challenge the metacognition, challenge the mathematical reasoning, and to let them have the deep learning and understanding of math. And third, we actually haven't yet touched with it. So the formative assessment should be integrated with the e-learning and the digital learning. Like the iClickers and iCloud, a lot of the new products trying to infuse the formative into the digital classroom. And FEM prototype. So this is a graph as show the feedback loop to see formative actually infused into each step of this learning from the in between of standardized score and impact the teachers' teaching goals, centers on the students' learning, and finally we come up with some professional learning community. So that is a scenario. Maybe you cannot really imagine understanding that the teacher giving test. It's not the test just all the students knock their hand and writing up on papers. Teacher going into the classroom, interact with them, and the students still need to hands-on reply to the teacher's questions and prove they understand and show their work. So we use this prototype FEM prototype concept and created this PT module. It's an online training module. So with three steps of the training module, first the teachers learning about the rubrics, learning about using the materials, and the tool or instrument we created to teach the kids and assess them. Second part is focused on the student learning. So after they gave the interim test, which is a very short one, to collect the data and figure out where is the students missing and monitoring their progress, they come up with the data. And then they go to reteach, use the data and use the information, and might go to the adjusted assessment, adjusted curriculum and reorganize those units. Between each of the training session, we give them some evaluation forms, which is the green part. So it's like a checkout list and the evaluation form to prove the student, to check out if the teachers have fully compliant with the rubric. So I'm going to skip into the result part. That's most parts that a lot of the stakeholders and principals want to say is the trend map. So on the trend map into 3 index, we can see that in a very short, like less than 1